I think this is a really exciting topic. It was one that was suggested in the survey that we sent to the GCC TI members last year. In response to that survey, there were a number of folks that suggested that we have one of these meetings that focuses on AI and machine learning. And I can tell you also that from grant reviews that I've done and from manuscripts that I've been reviewing over the past years, this is a topic that is really, coming to the forefront. It is something that we all need to be thinking about. It's a technology that we're all going to have to engage. And so we've sort of set up this this symposium as sort of an introduction and a discussion of the potential for this technology and to bring everybody up to speed on where we are with AI and machine learning in imaging. So we've got a great lineup of speakers set up for you today. Robia, do you have anything you'd like to add to the welcome this morning?
No, just I definitely echo the sentiment. We have a great range of speakers covering a variety of topics, basically from machine learning and AI 101 to applications to people who really are driving the science of the AI and the machine learning. So I think with this, and many different applications to many different types of imaging, not just MRI, which is near and dear to my heart and Jim's heart, but hopefully through this meeting, people will learn a lot and hopefully develop some new collaborations and ideas.
Okay, and Suzanne, could you take just a minute to tell us a little bit about the GCC?
Absolutely, happy to do so. So I'm Suzanne Tomlinson. I'm the Director of Research for the Gulf Coast Consortia. For those of you that don't know, this Translational Imaging Group is a GCC cluster, growing, been going for a couple of years now. This is their second conference. And so I want to just tell you a little bit about, okay, hang on, there we go. A little bit about what we've got going on. For those of you that don't know, the GCC is a collaborative network of 7 institutions in the Houston, Galveston area. These are our full members. We have the presidents from all 7 of these institutions came together back in 2001, actually. IBT came on a little bit later. But they have decided that it's really important to come together and build training and research programs that are better together than any one institution could do on their own. And so through that, we've developed several of these consortia clusters or just newer groups. These are all different topic-focused research groups ranging from 100 folks to some of our larger ones that have over 700. We also have several different training grants. These are multi-institutional. Some have 7 institutions. Some of them have at least 3, but they're administered through the GCC. And then we also have through the GCC an MOU for shared equipment and resources, as well as work with our institutions for new equipment and resource acquisition. This is just an example of some of the shared equipment that we've gotten through the GCC for anybody who is doing any kind of drug discovery and development. We have a network of cores that has gotten all of its inaugural funding from CPRIT, so there's certainly a lot of cancer research going on, but also some other research in these cores as well, so keep that in mind. And then finally, I just want to say a couple of things about a seed grant. For those of you that don't know, we have a seed grant that's administered through the GCC, awarded. We're in our second 10-year award from the John S. Dunn Foundation. This is the Collaborative Research Award. And sorry about that, my slides are disappearing. This is the Collaborative Research Award. We'll have the new RFA will go out on March 1st of 2020. What we want for these is brand new collaborative teams with members from at least 2 different GCC member institutions. Those pre-proposals are due within your own institutions by April 1, a full proposal by June, and basically they're $100,000 per team. So we just hope that you'll keep that in mind and you know, participate with this research consortia throughout the year. They've also got, I'll tell you, this imaging group is sponsoring a hot topics in imaging workshop that takes place on the third Wednesday of each month at 1. So if you're interested in that, you should shoot us an email so we can add you to that list so that you can be made aware of those. Of course, those are all taking place by Zoom right now. But they've been really exciting. We have one coming up this next week. And yeah, so please feel free to reach out to us if you have any questions about that. And with that, I'm going to go ahead and turn it over to Robia, who can introduce our first speaker.
Okay, great. Thank you, Suzanne. So our first speaker is Dr. Jia Wu. He's comes to us from MD Anderson. He's a new assistant professor. And we're very fortunate that he's willing to give us basically an introduction to artificial intelligence and medical imaging, as that is his specialty. He does a lot of applications with machine learning and oncology. So we're very excited to have him give us this introduction to start off our symposium. So Dr. Wu, please.
Thank you so much, Robia. And thank you for this great opportunity to present the AI 101 in medical imaging and open the door to discover clinical meaningful imaging markers in precision oncology. I joined MD Anderson a couple months ago and building a lab that focuses on using AI to extract meaningful features from radiographic and histopathological data. And the purpose of my talk is provide you with a general, high-level AI overview to start analyze this rich imaging data. And hopefully after my talk, you will be motivated to start contribute to this exciting research area. The holy grail of precision medicine is to tailor optimize the treatment plan for individual cancer patients. And compared with one-size-fits-all approach, a precision medicine accounts for personal level variability. And in current clinical practice, and this personal level variability mainly relies on the measurement from the tissue samples. However, the tissue sample omics data alone is not sufficient to stratify the heterogeneous cancer population. And one solution here is to start investigate medical imaging. Compared with molecular analysis, imaging actually have many advantages here. It can provide a more robust global characteristics of tumor and its surrounding tissue. And also the recent technology advancement have significantly improved the imaging quality and capability. Moreover, some novel molecular imaging modalities allows us to non-invasively profile the biological activities of the tumor and also its longitudinal changes. Because of these standardized and quantifiable imaging characteristics can be extracted. And by adopting the AI into this medical imaging field, we can extend the imaging's role in the era of precision medicine. There are plenty of exciting AI projects going on. I'm sure you will hear some of them today, aim to improve the efficiency of radiologists' workflow, including diagnose and staging and response monitoring and prognosis. This VM plot clarifies the relationship among deep learning, machine learning, and artificial intelligence. And in medical imaging analysis, machine learning algorithms are widely used to identify useful patterns within the heterogeneous imaging data. Inside the machine learning, there are three main categories known as supervised learning, unsupervised learning, and reinforcement learning. And supervised learning relies on the labeled data to fit the model. By contrast, the unsupervised learning can detect the intrinsic patterns from this unlabeled data. The reinforcement learning in general learns from its own mistake to reach the optimal performance. And the majority of the AI projects use the supervised and unsupervised learning. Let's take a close look at the difference between supervised and unsupervised learning. And supervised learning is where you have this input variable X named as features and an output variable Y named as label. The aim here is to build an algorithm to optimize the mapping function from the feature space to the label space. By contrast, this unsupervised learning is where you only have the input features X and no corresponding output label are available. The goal here for the unsupervised learning is to model the underlying structure or distribution in the data in order to learn from the data. And these are called unsupervised learning because unlike the supervised learning where you have the label to guide the learning process, but here rather the algorithms are left to their own to discover and present the interesting structures in the data. The quantitative medical imaging analysis procedure is generally known as radiomics. And this figure shows you a conventional radiomics analysis pipeline. First, when we will curate a relevant imaging population, and then we are going to extract the structured features X from this raw imaging unstructured data. And this step will include segment out the tumor, the regions of interest. And then we will derive different types of imaging features to characterize the tumor phenotype from imaging scans. And then the last step is to build advanced classification or regression algorithms to link this feature X to the clinic endpoint Y. I'm going to show you one of the first example of supervised learning where we apply the radiomic concept to study FDG PET scans and to predict the risk of metastasis in lung cancer patients. First, let me give you some background. For early stage lung cancer patients, a stereotactic ablative radiation therapy is a standard procedure that induced to eliminate the cancer cells. And the radiation treatment has excellent local control. However, the distant metastasis is a major failure pattern here. Since no lymph node biopsy is performed, clinically there's no reliable way to predict the individual patient's risk of metastasis. And here our aim is try to test if the PET imaging scans can be used to identify the subgroup of patients with high risk of metastasis. Therefore, these high-risk groups will be administered with adjuvant chemo after their radiation therapy to prevent the distant metastasis. And by contrast, the low-risk group patients can be spared from this chemotherapy and its associated side effects. In order to do our analysis, we carried a radiomic study with supervised learning since we have the patient's imaging data and also we know their outcome label Y. Here comes the overall study design. And we collected a pre-treatment PET scans of 100 patients from Stanford Cancer Hospital. And the cohort here was divided into training and testing groups based on their year enrolled into the treatment. And within this discovery group, we extract a set of 70 features. And on the left side, you can see the detail of these 70 features to measure the tumor shape, the tumor texture, intensity, and some filter-based texture features. And then we test the robustness and the redundancy of these imaging features. After that, we apply the feature selection algorithm nested with a Cox regression model to predict their risk of metastasis. And more importantly, this build radiomic model was independently validated in this holdout testing cohort. And here comes the model performance during the discovery and the validation cohort. And this first column shows the radiomic, final radiomic models, which selected two imaging features. One is the PET SUV intensity feature, which reflects the metabolic activity. And another is the texture feature of SUV map, which reflect how this SUV intensity distribute within the tumor, measures this SUV heterogeneity. And by contrast, we compare with the standard clinical factors, including tumor volume and SUV max, and they all feel to be informative during the validation. And to summarize this study, and we have carried a pilot radiomic analysis with supervised learning idea to study FDG PET and approved its feasibility to tailor treatment plan for lung cancer patients. With the advent of lung cancer screening, more and more early stage lung cancer patients will be detected and treated with radiation therapy. Therefore, this algorithm will potentially play a key role to tailor the patient's treatment plan. However, there are several limitations here where we only characterize the primary tumor from the pretreatment scan. And in the future, to better characterize these cancer patients, we may need to quantify both tumor and lymph nodes from available longitudinal scans. And in a recent study in head and neck cancer, we adopt this idea and start to extract the radiomic features from both tumor and lymph nodes based on the pretreatment and the mid-treatment slides. Through doing this, we approved the prediction accuracy can be further improved. And as you can tell, this analysis is not fully automated. For example, the radiologist need to segment the tumors, and we also need to handcraft the features and tune the machine learning algorithms. In the future, a more exciting opportunity is to automate this whole process with newly emerging deep learning algorithms. This slide on the bottom shows the structure of a standard convolutional neural network. And due to the time constraint, I will not be able to elaborate the details of the deep learning. And in brief, the first half of this neural network is based on the fully convolutional layers. It is similar to the feature extraction procedures we have done in the radiomic analysis. And this through repeating the convolving and pooling operators multiple times, it can distill the features from a localized area to more global concentrated area. And the second half is the stack of fully connected layers, which can predict the clinical outcome from these given features. And one key here is through the loss backpropagation, this convolutional neural network can automatically tune these hyperparameters to reach its optimal end-to-end prediction. And a robust deep learning algorithm will rely on large, well-curated and labeled dataset, as well as great computational power. And let's summarize what we have learned so far from the first example. The first point I want to share is training the machine learning models, as you can tell, will depend on the person's favor and empirical knowledges, such as what type of the features to extract and what algorithm to use for feature selection and which machine learning algorithms to use. And secondly, the key here is to validate these trend results. In the training results, theoretically, we can reach perfect prediction results. And we need to prevent the information leakage during the testing and also rigorously test this generalizability of these trend models. And essentially, we are balancing between the overfitting and underfitting. And there are several key points I want to remind you here. So the similarity between the training and validation data sets will affect the model performance. And also the training and validation compared with the general population out there will also affect how generalizable your trained model will be. And how these models interpretability will affect its adoption in the clinical setting. And also we need to rigorously evaluate the model's performance in the clinical context to compare with what's already known in the clinical settings. And also we need to know no model is perfect. We need to acknowledge its limitations. Next, I want to switch the gear to show you another example where we leverage the unsupervised learning algorithm to address a very interesting intratumor spatial heterogeneity question. And recently from this biology study, we have found that tumor actually is not a homogeneous entity. Instead, it contains many distinct subclones. And this subclone will corresponding to different gene profiles. And we hypothesize that certain regions with a tumor can be more aggressive and treatment resistant. And since the imaging provide a global view of the tumor, and the habitat imaging aims to uncover how these subclones manifest on these medical imaging scans. And these subclones are named as habitats. And since we don't know how many habitats exist, that we don't have the labels to train these models. So it fits well to adopt this unsupervised learning. And conventionally, there are several ways to define these habitats from the imaging. One is with predefined threshold, such as using SUV value to define the metabolic tumor volume from the PET scans. Or another way we use is based on the tumor morphology, as shown on the right bottom figure here, where we can define the pre-tumor and the intratumor area. However, these existing two ways to define the habitat imaging have some weakness. There are certain imposed rules to define these habitats, which can cause some uncertainty to analyze multi-center data. However, another more important point here is they all implicitly assume there's only two habitat regions exist, which may not be the truth. In order to explore these habitat regions, which is generally unknown, we address this challenge with unsupervised learning. And this figure shows the general idea behind our pipeline. And the task here is to automatically identify how many habitat regions exist and automatically segment them out. And first, based on these given pictures, we over-segment it into many superpixels. And each superpixels contains perceptually similar pixels, and where, in other words, pixels with similar color and textures are grouped together. Then by treating each of these superpixels as one sample, we run a second level, population level clustering to identify how many different groups of superpixels exist. And through doing this, we identified there are two types of the superpixel existing. One corresponding to dog, another corresponding to grass. By projecting these clustering results back to the superpixel space, we can automatically segment this whole image into these dog habitats and grass habitats. And similarly, we used this concept as a knowledge discovery approach to explore the intrinsic habitat regions in this cancer field. And given these multimodal imaging scans, we first over-segment the regions into a large number of superpixels. Then by treating each of the superpixel as a sample, we run a population level clustering to identify the number of the habitats as well as the membership of each of these identified habitats. By projecting this population level clustering back, we can consistently partition the tumor into these identified habitat regions. We call it habitat maps. And these habitat maps serves as a new way for us to quantify this intratumor heterogeneity. And I'm going to show you how we adopt this unsupervised learning idea to study a clinical challenge in breast cancer field. And here we adopt this habitat imaging to address the clinical challenge here. is to extract perfusion MRI patterns that can predict tumor recurrence after surgery. And first, at the patient level, we, based on these multi-parametric perfusion maps, we partition individual cancers into a large number of superpixels. Then at the second stage, we pull in all these superpixels from every patient to run a consensus global level clustering to identify the number of the habitats, as well as consistently partition each of these tumor into its intrinsic habitat maps. And given these habitat maps, we find out consistently there are 3 habitat regions corresponding to these low, moderate, and high perfusion areas within the tumor. And given this habitat map, we propose this multi-region spatial interaction matrix to specifically characterize the spatial interaction and arrangement of these reviewed habitats. And in general, we iterate through all these tumor voxels and to query its neighborhood. For every tumor voxels, if there's, for example, if there's a blue voxel has a red neighbor, we'll add the entity to this corresponding co-occurrence matrix and summarize its distribution and interaction in this habitat interaction matrix. After we extract this habitat related interaction matrix, we proposed a network analysis, which is also an unsupervised learning algorithm to explore the similarity between these discovery patients and to discover the clustering patterns based on this discovery cohort from UCSF of 60 breast cancer patients. This left side shows a sparse graph, and it indicates the topological relationship of these 60 training patients, and each of these vertex represents the 1 breast cancer patients, and their distance mimics the similarity in the feature space. And after the spectrum clustering, we identified 2 groups within these 60 patients, and we colored them in red and green. And we tested their survival, and we found they are significantly different in terms of the recurrence-free survival. In average, as you can tell, the red group is 3.5 times more likely to generate the recurrence compared with the green group. And we further validate our identified imaging analysis patterns subgroup on this multi-center clinical trial data. And similarly, we extract these habitat maps and related interaction features. And then given these features, we embed these testing patients into the training network, so the training labels can be propagated to this validation cohort. And as shown here in these Kaplan-Meier curves, the habitat features still remain significant to stratify the overall patient's survival as shown in the figure A, as well as the subgroup ER positive groups. Similar trend was also observed for HER2 positive group. And also another interesting finding here is in the original I-SPY study, the researchers find out the pathological complete response is a strong prognostic factor. Here we have shown that for patients who did not achieve the pathological complete response, the habitat imaging features can further stratify their recurrence-free survival as shown in this figure D. These results indicates a potential synergy between habitat features and the pathological findings. And in the end, we want to explore what we have discovered by using this unsupervised habitat imaging. And we observed that patients characterized as high risk of recurrence had generally a large fraction of low perfusion subregions and more interactions between low and medium perfusion habitats within the background parenchymal. We hypothesize that these imaging patterns of aggressive tumors could be driven by a hypoxic microenvironment within the tumor and at the invasive margins. And in the future, these findings need to be prospectively validated in order to make real clinical impacts. And moreover, in the future directions, we can combine this dynamic contrast enhanced data with other MRI imaging modalities, including T2 weighted and diffusion weighted imagings to better define these habitats. And here, we only analyzed the primary tumors at baseline scans. And in the future, we can analyze the longitudinal scans and to extract how these habitat evolve under the therapeutic pressures. And hopefully it can improve the prediction accuracy. And we are now extending this unsupervised habitat imaging analysis to study the lung infection patterns of COVID-19 patients. And specifically, we hypothesize that beyond the known subregions read by radiologists on their daily practice, such as the GGO, the consolidation area, and habitat imaging can serve as a knowledge discovery approach to start identify subtle yet significant infection regions and their interaction patterns. Beyond using the radiographic scans to predict the clinic outcomes, the treatment response, there are ongoing research efforts very interestingly to correlate the radiographic imagings with molecular omics data. And this imaging and genomic correlation study is generally known as radiogenomics. On one side, the top-down design allows us to build imaging models as non-invasive surrogate, like virtual biopsy, to predict interested molecular markers. And on the other side, we can reverse this top-down design in order to identify the underlying biological correlates associated with specific imaging patterns to validate its biological meanings. And there are plenty of opportunities to adopt AI algorithm in radiology field. Let's look at the cancer development timeline here. Cancer is a gene disease start from random mutation and evolve towards the detectable lesions. And in clinical practice, the imaging is used to diagnose cancer patients, where AI can be used to minimize the false positive recalls. And in patients' staging and treatment, the AI can be used to optimize the therapeutic regimen. And after that, the imaging is used to monitor and follow up the patients, where we can use AI to balance the treatment effects and quality of life. And even before the lesion is detectable by diagnostic scans, for the general screening population, such as in breast and lung cancer field, it's feasible to adopt AI to start investigate the abnormal imaging patterns and allow early cancer detection and prevention. And medical imaging contains rich data that can be mined for personalized medicine. And parallel to this radiologist expert eye to read the scans, we can start to adopt these various AI pipelines, including radiomics, habitat imaging, deep learning, to start extract quantitative imaging features. And more importantly, we need to harness this rich imaging data to improve the diagnostic, prognostic, or predictive accuracy. And another research direction here is to decipher the mechanisms behind imaging patterns through radiogenomic analysis, and where this imaging can be correlated with the underlying molecular omics data. And alternatively, combining the imaging with molecular data could potentially improve the prediction accuracy. And now we are barely scratching this challenge, the surface of these challenge problems here, and future research efforts are critically needed in order to translate these into clinical practice. And in order to make real clinical impacts here, there are several challenges we need to overcome. including how to harmonize this data and how do we construct these models and validate these models. And this also provide a great opportunity where we can start to integrate this multidisciplinary knowledge to solve and address these challenges. And to wrap up my presentation here, in the clinical practice, we are now start to extract multi-scale information about cancer patients, including the radiographic scans, histopathological slides, as well as these advanced molecular measurements. And the AI actually provide us a great opportunity to start extract this meaningful information from this unstructured data at the individual patient's level, and more importantly, to build the pipeline to integrate this information towards precision medicine. And my lab is looking for talents to join and work on these exciting projects. So if you are interested, please send me an email. With that, I will close my talk. Thank you for your attention.
Great. Thank you so much, Dr. Wu, for an outstanding presentation. That was excellent. I'd like to open up for a few questions. I think we have just a couple of minutes from the audience. Anybody have any questions? I actually had one just thinking about it. It sounds like the way you described it, it sounded straightforward, but in reality, it sounds like the iterations of building the model and then validating the model could actually be quite challenging and time consuming. Do you have comments on that?
Yeah, yes. So I would say, there are two approaches generally in my mind here. One is like a purely data-driven approach, and including like radiomics and deep learning, where we let the data tell us the patterns or information from these patients. Another approach is more like the habitat imaging, where we have these very well designed biological hypothesis. And then we start to, along this line, to design, handcraft all these features. And there's no better or worse, right or wrong. I would say this is based on the different problem, maybe have more suitable tools to use. And in terms of the validating the model, I would say maybe share the model to validate in another independent cohort. That would be the gold standard. And also another thing to keep in mind is how similar these patients are, because the cancer treatment now are evolving so fast in certain type of the cancer. So make sure the patients all have similar demographic characteristics, similar treatment, and also the high quality follow-up data. And another thing I want to mention here, highlight, is for the imaging protocols. I'm sure a lot of experts in that field, how this imaging protocol, how do we image these patients, what type of the machine do we use, that will also affect how the imaging features will be extracted. Okay. And luckily, there's already many researchers there to use the advanced deep learning to harmonize the data. So that can be another thing to add on this existing pipeline to make it more robust, to generally applicable to many different sites.
Okay, great. Thank you so much. We do have to move on, but you do have a couple of questions in the chat. So if you wouldn't mind going to the schedule and clicking on your talk and answering the questions in the chat, we'd appreciate it. But we do have to move on to the next speaker. But thank you again for an outstanding presentation. That was great.
Thank you.
Okay. So next we have Dr. Shella Kyle-Holtz. She comes to us. She's a newly minted full professor from my understanding. Congratulations, Dr. Kyle-Holtz. And she comes to us from Georgia Tech and Emory. And let me click on hers. She is internationally recognized for her efforts in resting state functional MRI. She just does some absolutely brilliant work. And in her presentation, she actually uses AI and machine learning in her resting state and functional MRI studies. So this is an applications presentation. So with that, Dr. Kyle-Holtz, we're very grateful that you're willing to be with us here today to share your work. And please go ahead. I think you're muted.
Yeah, there, how's that? I had to find the button once I shared the screen.
Okay, perfect, thank you.
Thank you so much for that kind introduction, and thank you and the other organizers for giving me the chance to come and talk to everybody today. I'm really excited about this collision between machine learning and neuroscience, and I think there are so many cool projects that could come out of this. So I'd like to kind of offer some of the descriptions of things that we've been doing just to get you all thinking about, other ways that we could use this to answer questions that we haven't been able to answer before. There we go. Okay. So we know the brain is a complex system. It has activity occurring at many different scales, starting at the scale of the single synapse in milliseconds, right? So meaningful activity occurs at that level. We have areas organized into layers and columns that share common inputs or common functions. We have these neural circuits that have been the subject of many neuroscience studies where you're looking at kind of feedback between the thalamus and cortex, for example. We have these functional networks that you see all the time in these fMRI studies where different parts of the brain kind of coordinate their activity to do a particular task. And then we know that there's widespread neuromodulation from the systems that keep us awake and paying attention during these virtual conferences or let us fall asleep when things get a little bit too boring. And mixed in with all of this, we mostly think of neural activity, but there's also a lot of glial activity. And the astrocytes in particular may be important for some of these functional imaging modalities. But all of this signal is going on. Things at the very localized short time scale level kind of add up to make these levels of activity that we measure across the whole brain. But at the same time, the whole brain patterns of activity can also constrain whatever happens at that very localized level. So there's this really interesting feedback that makes the brain's complex system really exciting to study and difficult to try to tease apart the different components. Now, with all that's going on in the brain, what we get out of it is whatever we can observe with imaging modalities. And there are different approaches. We could get EEG or MEG, but if we do that, we've got great temporal resolution. We're kind of limited to the surface of the brain and have poor spatial resolution. On the other end of the scale, there are these optical imaging techniques that are letting us image many, many neurons at a time now, but usually only from the surface of the brain and only from animal models. So if we want to do this in humans, really our only good option right now for whole brain, high spatial resolution coverage of neural activity is resting state fMRI. And so by resting state, I mean, we're looking at spontaneous fluctuations in neural activity rather than changes that are specifically linked with any given task or stimulus. So this complex system of the brain has all been kind of condensed down into these low frequency MRI signals, but we still have a ton of data. We have tens of thousands of voxels, we have thousands of time points, and we need some sort of method to make sense of what's going on. So the traditional approach has been to look at something called time averaged functional connectivity. And you can really think of this as correlation analysis. The simplest implementation of it is to take a seed somewhere in the brain and take the average time course from that seed, let's say maybe right somatosensory cortex, and we average all the voxels in that area, we get a time course, and then we do correlation between that time course and everything else in the brain, with the goal of finding other areas where activity is highly correlated. Now, if activity in two areas is correlated, it kind of goes up and down together. And so that we assume there must be some sort of coordination of that activity via communication, through these neural circuits. And so we call that functional connectivity. Now, when we talk about applying this in a whole brain way, a lot of times an approach that's used is to parcel the brain into a bunch of different areas. And I'm showing one parcellation from the Human Connectome Project over on the left. And those parcels are based on similarity in function, similarity in anatomy, all of these different things that they use to kind of define functional areas. So we can take that parcellation, divide the brain up into bits, And then we can make a matrix like the one I'm showing on the right, where we get the functional connectivity between all the different pairs of parcels in the brain. And so let's see, I think you can probably see my cursor. And so on the diagonal, we have the correlation between each area in itself, which is 1, of course, so that's that bright line. And then each of these squares represents a network because we've kind of organized these parcels into networks for better visualization. And so you can see in the squares along the diagonal here, we have high correlation within networks. And sometimes we also get high correlation between networks. Like here, this is correlation between the visual network and the dorsal attention network. Or maybe anti-correlation between the task positive networks and the default mode network. But that's the kind of summary that we get out of these time average functional connectivity measurements. So clearly we're throwing away a lot of information. We have maybe 100,000 voxels for this HCP data. We have 6,000 time points per subject. We have 1,000 subjects. We're taking all of this and condensing it down to a 360 by 360 matrix because we have 360 parcels for this particular parcellation. So, you know, there's a lot of, that's useful, but there's a lot that we're missing out on here. For example, we know that the brain's functional patterns change over time. While you're in the scanner, you're not doing the same thing the whole time. You know, maybe you're laying there and you're like, I'm getting bored. I'm tired of being in here. This is uncomfortable. I wonder when lunch is going to be. And then maybe suddenly you remember, gosh, I forgot to stop and pick up, you know, the butter that I need to cook supper tonight. So your brain is doing different things, but we're averaging all that together to get these sort of metrics. We also know that your level of vigilance is changing over time. And, you know, I'm a great example of this because the first thing I'll do if you stick me in a scanner is fall asleep. And that's pretty common. And so people in the scanner are getting drowsy, kind of waking up again, and, you know, generally kind of drifting off over time. We know that these types of variations in functional connectivity and in the state of the brain also relate to behavior. For example, if you look at the anti-correlation between the default mode network and the task positive network as a function of time, the state of that anti-correlation predicts how fast you react on some very simple tasks. So overall, we need something that captures some of this time dependence in order to look at the dynamics of the brain as a complex system. So there are a lot of different approaches for doing this, and I'm going to go through these very briefly. For example, you can do a sliding window correlation where you take your time course and instead of doing correlation through the whole time course, you take a window that you either slide along the time course or you segment the time course into windows and get some sort of timing information there. You can do these co-activation patterns where you look at whenever individual areas pass a threshold and call those active and everything else inactive. And both of these are typically followed by clustering, which then gives us kind of groupings of common patterns that we see over time that we might call brain states. Now, the one I jumped over is quasi-periodic patterns. I want to say just a few more words about those because you're going to hear more about that later in the talk. So quasi-periodic patterns are repeated spatiotemporal patterns. So not just brain states, but brain states that evolve over time. And the one we see most dominantly in the human is something that evolves from default mode activity with negative activity in the task positive mode to a reversal of those two and has propagation along the cortex. The whole pattern takes about 20 seconds. And it recurs a lot in the background of whatever else is going on in the brain. So when we do a pattern finding analysis, we get a pattern template of that. And that's this shown over here on the right. It is a 4D pattern, volume over time, the 20-second period. And then we have a sliding window correlation vector, which tells us how strong that particular pattern is at any point in time in the scan. Okay, but for now, we can ignore that because I want to go on and talk about a different way to look at, you know, the dynamics of the brain using machine learning. Partly because we were inspired to do this, partly because we know that from simulations that if you look at, you know, something that's closer to the raw data, not filtered data, not sliding window correlation, you tend to get better performance with our dynamic analysis algorithms. And so we saw a talk by a colleague at Emory who was using this sort of approach to look at behavior in fruit flies. And we said, yes, that's what we need. We need to look at brain behavior. So we've kind of adapted his dimensionality reduction approach to look at how the brain activity evolves over time. So we assume that every scan, every time point of every scan is one time point in this highly dimensional space defined by the values of activity at all the different voxels. And then we do this t-distributed stochastic neighbor embedding, or I'll also call it t-SNE, which is a machine learning algorithm that uses gradient descent to minimize the divergence between the distributions for your high dimensionality probability of what different voxels are close together, what different states are close together, and the low dimensionality representation of that. So it kind of learns this 2D or 3D representation of this high dimensional space. We applied this to data from the HCP. We had 2 times 7, so 14 task scans plus 4 rest scans from, at the time, 446 volunteers. So these are the tasks that were included. We threw the task in with the rest. I'll show you why in a minute. And when we took that data, we first reduced the spatial complexity by taking only 50 ICA components. And that's because there's a lot of redundancy in resting state fMRI. Voxels that are right next to each other tend to be correlated. We also expanded the frequency representation by using the continuous wavelet transform. So that took our time course, and instead of a single time course, gave us time courses at different frequencies over the course of the scan. And we fed all of that into this t-SNE algorithm. So here's what we got. I've separated out the task and rest here. We actually did the analysis together, so you can imagine overlaying them all for the total view. But one of the things we were interested in terms of this idea of brain behavior, was whether the brain at rest in the scanner, so when we're just looking at spontaneous activity, if it's actually different from what you see in the task. Because you are kind of doing tasks while you're in the scanner, they're just undirected tasks. You might be remembering something, you might be planning something, you might just be sitting there thinking about how uncomfortable you are and how much you wish you could itch your nose right then. So these are all things that we might do as particular tasks. So we wanted to look at the overlap between the states that we saw during the resting state scans and what we saw during the task scans. And if you look over here at the left, you can see that the rest scans kind of localize, this is a heat map now, so high probability are these yellow areas. And you can see that they kind of localize to the outside of this ring, and that the task tends to localize to inside of the ring. Right here, we've collapsed all the different tasks together, so there's no distinction between what type of task. So you do see some probability of finding particular task-related states during rest, but for the most part, we see several distinct states during rest that are not present during task, which implies that there's something different about rest fundamentally compared to task. So we can also then do a watershed algorithm, which basically treats these heat maps as representations of peaks. So high probability would be like a mountain peak, and we can draw lines around those peaks based upon where water would drain off from these, which is why it's called the watershed. And so we can segment this space very nicely into different brain states if we want to. We can also measure the velocity at different points in space and see how quickly people tend to move in or out of a particular area. And so over here for rest, this yellow is a high velocity, red is much lower. And so you can see when you're passing through the middle here of this low dimensional space, you tend to move very quickly. So people don't linger here, which is in accordance to the way we see these lower velocities and people staying in particular states during rest along the edges. So once we define a state, for example, this watershed area highlighted in pink, or this other one also highlighted in pink, we can then look at what's going on in that particular state. Oops, sorry about that. There we go. Okay. And so what happens is that if you look at this particular state, you can see that, activity is fairly flat. There's a little bit of kind of a low signal in these very low frequencies in particular networks. I should say that the horizontal axis here is activity in each area or network, and this other one is in each frequency band. So it gives us a snapshot of the power in different frequency bands in different areas for that state. Whereas if we come down to this other state, you can see that there's a lot of power in the high frequencies in many different networks, but there's some negative power, or lower than average power in the higher frequencies in those networks. Okay, so you can also take this and break it down even further. So here's my conglomerate of rest and task and everything that I mentioned before. And if you look at how different tasks map onto this, you can see some tasks like emotion require a bunch of different states, and others are like this gambling task are much more localized to very specific areas, the motor task. And each of these tasks has its own unique representation. We also looked at rest separately for the first and second resting state scan, and weren't able to distinguish between those two, which implies that they're, the states that are produced are not necessarily a function of you, falling asleep in the scanner, because then we would think maybe that second scan would have more of one particular state than another. Okay, so digging in then a little bit deeper, we can even look at how the brain moves around in this space during a task. So this task, this is work by Jacob Billings, and Jacob has broken it down into two different pieces. So it's a task where you watch a swarm of dots. And in some cases, the dots are moving randomly. And in other cases, they look like they're doing something coordinated. They're kind of moving together. And that's what he's calling mentalizing, because he pictures the dots as being, communicating mentally, I guess, to be able to coordinate. So you can see these have slightly different maps in this T-SNE space. And if you look into, their patterns of activity at that state, things are very similar in the high frequencies, but we get a divergence in the low frequencies. That really is what distinguishes, these random dots from these coordinated dots. And what I thought was really cool is that you can look at how the state evolves over the performance of the task. And so at the bottom here, each of these frames is a time point. And so you can see that, the state tends to move in this low dimensional space as a function of time while people are performing these tasks. And so this was, something that hints at brain trajectories, or this idea that there are particular pathways that activity likes to move from one configuration to another. So I'm going to show just really briefly some very new work from the lab. I got these slides yesterday. And what we're doing now is, instead of T-SNE, using an autoencoder approach to try to define a latent space where we can find brain trajectories. An autoencoder is a machine learning approach that is often used to denoise a signal. It basically takes a signal, projects it down into a low dimensional space, and then brings it back out to try to reproduce the original signal minus the noise. But anyway, we're looking at the latent space here because we thought that might be the best place to look for trajectories. And so this is a set that has two latent variables, and you can see this cycle. There's a circle that lasts about 36 seconds for this whole pattern. And it lines up well with that 20-ish second QPP that I told you about before. If you look at the main part of the cycle, we have activation in the default mode network, followed by progressive activation into the visual network, and then into the somatomotor network, and back to the default mode network. So I think this is really encouraging. And Xiaodi has gone on and we thought maybe the two latent variables were not enough to really distinguish these trajectories. So he's done something similar now with 8 latent variables, and he's seeing some of the same things. So here again, anytime this trajectory over here is red or these dots are red, that means default mode network's dominant. I think green is visual network and blue is somatomotor. And so you can see there, it's like, you know, there's a probability maybe for things to move from one type of network domination to the other. We're still trying to figure out the best way to characterize this. We've got some ideas, but would love to chat if people have, you know, input for this. Okay, so now I want to jump to a different way to look at the brain. And this is to try to understand the brain as a complex system using a model. There have been a lot of people who have worked on this idea of making brain network models. And the idea is that you take that parcellated cortex or parcellated brain I told you about, you use diffusion weighted MRI to map the structural connections and define how strongly each parcel is connected to every other parcel, and you make a network model like so. Now at each of those nodes, you put some type of neural mass model, and that's just a set of differential equations that describes how activity evolves. And so for this particular case, I'm using the Kuramoto model, which are coupled oscillators, but we've used many, many. Anyway, you let this go together, you let it run, and what you get out of it is simulated resting state fMRI data. So the thing about this is what we've learned is that many models and many parameterizations give the same average functional connectivity. So it's like any model fits our data. And that's not really informative if you want to be able to rule things out or to understand something about how the brain works. So I'm looking at this green column here. All of these models reproduce the same average functional connectivity. So the first thing we had to do is find a way to be more sensitive to things that could distinguish models that fit functional connectivity. So we went back to our time varying analyses. We found, we tried a lot of different things that I talked to you about before. Again, I'm only going to talk about QPPs to keep this a little simpler. And what we found is that time varying analysis does distinguish better between models, and that models that reproduce the average functional connectivity fairly well do less well on these time varying metrics. So here I'm showing, this is a simplified version of the QPP where each row now is the time course for a given area, and yellow means high activity, blue means low activity. So we're looking at a snapshot of the whole 20-second QPP right here over the course of the brain. Now, what you see in these brain network models tends to be something like this, where you get these chunky blocks and you flip between one state and another without this complex propagation that we see in between the two. And that's true whether we use, this is the firing rate model, here's a Kuramoto model. So this we thought was interesting and potentially very promising. We also found that the pattern depends on this coupling parameter. And so if you have a low coupling, you get something that's maybe a little less organized, and high coupling that starts to collapse into these two states. So we think these sort of dynamic metrics are going to be valuable. But now that we can distinguish between models and metrics, we still could use something to help us out, because you still have this huge space of parameters that you need to sweep for every model. So we thought maybe machine learning could help with that. So the first thing we tried was to learn the initial conditions. And so we implemented this by setting up a recurrent neural network. In particular, it was a long short-term memory network, which basically holds information from, you know, some of these prior time points and brings that as input to the current estimation. Anyway, we trained this, and we started out using the spiral data set where we have these green dots represent noisy measurements of the spiral trajectory, but we know the equation that generated the spiral, the underlying differential equation. And so what we tried to do was then to train this and then use just a few time points at the bottom, you know, the initial time points, to estimate the initial conditions, and then using the equation we know to find the rest of the time course. And it worked quite well. So because we want to be able then to maybe distinguish between models and parameterizations, we also tried learning the initial conditions and looked at what would happen if we learned the initial conditions with the wrong differential equation. You would expect that if, you know, we learned the initial conditions, we would probably start off fairly close to the right trajectory, but we would diverge over time. And that was exactly what we saw. These are the blue lines here, and here are our predicted values when we have the wrong differential equation. So again, that made us think this might be a useful tool. So we applied this to data from HCP again. And this is where things got a little bit trickier. So we're looking at the accuracy on the third time point. That just turned out to be kind of a good place to look before things diverged completely. And we looked at this as a function of global coupling strength. Global coupling strength tells us how much of a network influence we have at any particular point in the model. It is normally determined to be around 0.9 for our resting state data. Now, what's interesting is that you see that our accuracy actually goes down as our global coupling strength increases. And so it's lowest when we get to what we think should be the optimal value. So we thought about that, and it makes sense really, because we're basically putting constraints on our model, and this LSTM is doing better when it's unconstrained. So that's not really too surprising. But it's also not very helpful for us if we want to be able to distinguish between parameterizations. So the other thing we did then is we looked at if we perturb our known structural network, because the one that we've measured with DTI basically, if we start randomly reassigning edges to that, what happens as a function of this global coupling network parameter? And if we do that, basically if we go to low global coupling, we don't get any change. But when we go to optimal global coupling, we do start to see that our model can distinguish between better representations of the underlying network and poorer ones. So I think we still have a lot to do with this, but I think it's on the right track. The other approach that we took is to skip the parameter sweep, and instead of learning just initial conditions, to basically use our LSTM to fit the coefficients of the model at every point in our time course. So it's similar to before, but instead of learning the initial conditions and stopping, we learn the initial conditions, use our differential equations to estimate the next time point, feed the error back into the model, and continue with that until everything converges. And with that, we're able to get some really nice estimates of these latent variables for these different models. So I'm showing two models here, firing rate at the top, and the Wilson-Cohen model, which gives both excitatory and inhibitory activity here at the bottom. And so our output is in blue, our measured resting state fMRI is in orange, and you can see this track really quite nicely. And in addition to them tracking well, over here at the right are the latent variables that we get out. Now with the firing rate model, it's not really interesting. It's basically saying that the firing rate is proportional to the bold signal that we see, not a surprise. But this excitatory and inhibitory activity actually gives us more structure than we see in just the bold signal itself. And I think this could be very exciting if we can find a way to show that those values are actually accurate. I'm going to speed up a little because I want to make sure we have time for questions. I think I will skip this multi-time step prediction. It gets worse as we go along, but it does give us better estimates of average functional connectivity than even the brain network models. And it also maps to the QPPs much better than any of our brain network models did. So here is our empirical QPP again. And these are the ones estimated with the autoencoder. And now you're seeing some of that more complex structure that we would expect rather than this two-state solution that we get from the brain network model alone. Okay, so the downside of our, all right, I'll stick to the good first. Let me go to the summary and then I'll come back to the downsides. So I think machine learning holds a lot of potential for getting more meaningful information from these huge data sets. You know, I think data-driven approaches are our best bet for getting new discoveries. If we do, you know, analyses like we've always done, we're gonna find the things we've always found. And I think this also might make things a lot easier in terms of allowing us to reduce some of these time-consuming parameter sweeps, and more importantly, letting us sync models up to empirical data, which is something that's really been missing from this field of brain network models. And I think that the implications of that are going to be huge. Now, the bad is that, you know, when we just apply machine learning, we might not get interpretable data. And the downside I was going to mention about the approach with the LSTM, where we were learning the coefficients of the model as we went along, is that some of the dynamics can be captured by the LSTM itself. And so one of the ways we're trying to figure out is, you know, how do we make sure that the part that we're capturing with our model dynamics is actually, you know, what we're really wanting to get at, and not that the LSTM is holding the more important dynamics. I think, as always, when you're doing machine learning, you have to be careful with what you put in. If you put garbage in, you get garbage out. You put noise in, you get noise out. So I don't think machine learning for resting state fMRI is going to deal with our problems of, you know, motion, for example, in that patient groups have more motion than healthy controls, and that sometimes you can distinguish based on that alone. It's not a neural signal, but it's a signal. And I think we have to be careful about, you know, just throwing machine learning at stuff that we can do perfectly well with other techniques. So our future directions, I think what we really need to do to continue to use machine learning and neuroimaging and neuroscience is to tie it to something interpretable. Multimodal measurements, behavioral performance. As always, reproducibility is going to be an issue. And, you know, like I said, I think, you know, we shouldn't necessarily jump into machine learning for the sake of machine learning. You know, if a correlation is what you need, a correlation is what you should measure. But that said, I think it has a lot of potential to be applied in new and exciting ways. So I wanted to show one last thing before I open for questions, and that is what we're trying to do with these latent variables we've estimated from the model. So we now have estimates of excitatory and inhibitory activity. If we could measure those while we measured BOLD, we could validate our model. So we are currently in the process of setting up a method to do simultaneous optical imaging of excitatory and inhibitory activity while we measure MRI. And here is our first image of a rat skull through a fiber optic bundle that we're going to put in the scanner. So hopefully I'll have cool things to show about that in the future. So I just wanted to acknowledge everyone in the lab who's done the work, especially Amrit, who did the brain network models, and Xiaodi, who is doing the latent space trajectories. And then Jacob is now a postdoc in Czech, and he did all of the TSNE work. So this is an old lab picture. I have to put up our more recent lab meeting, which will probably look, you know, the concept looks familiar to everybody these days, social distancing and masking. So I'm sorry we don't get together in person for this conference, but I hope you guys will have some questions and some good discussion. Thank you so much for your attention.
Great. Thank you, Dr. Kyle Holtz, for that terrific presentation. Are there any questions for her from the audience? Please feel free to put them into the chat. I guess one question I have, Shella, just kind of springboarding of what, we have one. Okay, I'll ask my question later. The question is, how many separate dynamic states do you identify in a typical resting state functional MRI scan for healthy patients? Does that number increase or decrease for patients with cognitive issues like Alzheimer's?
That is a great question, and it's one I wish I really knew the answer to. One of the things that bothers me about this resting state fMRI is that a lot of times, you know, people will do some sort of analysis, a silhouette index or something, and come up with an optimal number of states, even for something like HCP data where we have tons of subjects, it's on the order of 5 states. And 5 states just seems tiny to me if we're talking about, you know, the amount of variability that's present in the brain. And so there's some concern that all we're capturing with dynamic fMRI might be these fluctuations in vigilance levels. But now when you're talking about comparing your healthy controls to a patient group, that becomes even trickier, because if you go by some, you know, objective criteria, you might get different optimal numbers of states for the 2 groups. But then when you look at, say, Alzheimer's compared to healthy control, you might find that they only had 3 states, but in one of their states, 2 of the healthy control states are kind of mashed together. So I think, you know, the only answer is to be specific about what you've done when you're trying to interpret it. So if you decide both groups are going to have 5 states, you might have to, you know, be willing to accept that 2 of your 5 states in the Alzheimer's patients might look more similar than they do in the healthy controls. And that maybe that should be interpreted as, you know, a lack of state variety. But I don't think any of those have really been settled yet. This is a big open question.
Okay, great. Let's see if there's any other questions. Suzanne, do we need to move on now, or can I ask a quick question?
Because we have a break, certainly feel free to go ahead and ask.
Okay.
And then we'll have a break.
Charlotte, what I guess I was wondering too is how well do some of these models transfer from preclinical to clinical?
That's a great question. You know, most of the brain network models have been implemented for humans. So it's almost a question of how well does it translate backwards. The problem though is in humans, nobody can really test this. So you can make models and you can say, oh, the hop bifurcation model reproduces more features that I see in resting state fMRI than the Wilson-Cohen model. But nobody can actually test those in humans. It's just about fit. And that has a lot of bias depending on what sort of parameters you've chosen. So I think we have to go to the preclinical models. And I think there are only a couple of labs that are really starting to do that, partly because we're just now getting the tools to be able to do these sorts of simultaneous multimodal measurements that we need to really be able to validate things.
Okay, so then I think that brings me to the question I was initially going to ask, which springboards off what Jim was saying. How do people new to the field get involved and start incorporating machine learning into their studies?
I think the first place is to have a question and to have a reason that the current analysis methods won't work, because that helps you design your approach. Now, you know, my group is by no means, you know, a group that's expert in machine learning. But I think what we are good at is taking what's out there and adapting it to answer a question that we want to answer. But I don't think it's that hard to learn to get to that level. So I think if you have a question that's really driving, you know, you're really motivating you to move into machine learning, that kind of, you know, it dictates its own answer. But it's great to reach out to experts. We have some great expertise here at Georgia Tech where people really, you know their machine learning inside and out, and they've helped us a lot.
Okay, that's fantastic. Well, I guess if there aren't any other questions, it is now time to take a break for networking. Thank you again, Dr. Kyle Holtz. This was a fantastic talk. I really enjoyed it and appreciated all that you had to share with us. And I guess, Jim, Suzanne, anything else you want to add, or are we ready for our networking break?
No, we can take a break and we'll reconvene at 10:40.
Okay, great. Thank you all.
Thank you all so much.
See you all back at 10:40. All right, well, welcome back, everybody. I hope you had a nice 15-minute networking and bio break. It's my great pleasure to introduce our next speaker this morning, Dr. Anant Anapragada, who received his PhD in chemical engineering from the University of Michigan in 1989, then took a couple of academic postdoctoral fellowships at the University of Minnesota and at MIT. Then he spent 10 years working in industry at Abbott Laboratories, Secuse, I don't know if I'm pronouncing that correctly, which was then acquired by Alza and then acquired again by Johnson & Johnson. So he spent a decade working in industry. He returned to academia at the Cleveland State University and Cleveland Clinic Foundation in 2000. Then he came to Houston to UT Health in 2003, joined Texas Children's in 2011, where he is now serving as Professor of Radiology, Vice Chair of Research, Director of Basic Research in the Edward B. Singleton Department of Pediatric Radiology. And he also has a joint appointment at Baylor College of Medicine. His research interests are in the development and translation of nanomaterials for medical and imaging applications. And he's spun off a number of businesses successfully that sprung from research that was carried out in his lab. And today he's going to present with us on translating AIs in pediatric radiology. Dr. Anapragada.
Thank you, Jim. This is one of the areas of active work in my lab. It's driven primarily by an assistant professor in my group, Zbigniew Staroselsky. Jesse, Ryan, and Nakul Reddy also contributed to some of the work that I'll talk about today. They were fourth-year residents in our department when they did this work. And my daughter, Akshaya, who at the time was an undergraduate at Harvard and is now at Hopkins, also contributed to this work when she worked in my lab. I do have to declare my conflicts of interest. Companies on the left that I founded that have an involvement in some of this work. Companies on the right that I wish I had founded, but I didn't. So let's talk about the pediatric radiology workflow, which only differs minorly from the adult radiology workflow. And I put this slide up mainly to talk about where in this process AIs could be involved. So, before the appointment, there's the referral process, and then there's a determination of scanning conditions. And I class these all as pre-appointment. And then the patient actually shows up for the scan. And then there's stuff that can happen when they're actually at the scanner or in the scanner, where you can optimize dose in the case of, shall we say, CT. You can optimize image quality by adjusting scan parameters. Probe positioning is a big issue, particularly for ultrasound, and sequence optimization for MR, obviously, and then reconstruction algorithms that are AI-based. And I would class all of those as scanner-based applications of AI versus everything that happens in the pre-appointment stage as part of the business process. And then there's post-appointment in the image interpretation, classification based on the image as pathological, normal. You could stratify grade. You could pick up incidental findings. And then there's the actual report writing, dictation, creating the right dictation template for that particular patient and that particular scan, filling the dictation itself, as well as the sign-off. And I call these PACS-based in the sense that applications of AI in this area, they're not on the scanner, they're not part of the business process. They're all in the radiologist's hands when they're reading the images. Most of the work in my lab has been focused in this area, in the interpretation area, and I refer to these as interpretive AIs. And of course, there are applications in all the other areas, but I'm not going to really be talking about them. I'm going to focus on a few things in my lab that have worked in this interpretive area. So how do you pick an application? Where can interpretive AIs help? And this is a heuristic that has been going through a fair amount of refinement over the last few years that we've worked on. So if you think about how much time a radiologist spends reading an image, the more time it takes, the more expensive that read is. Now, compare that with the reimbursement. How much do you get back? How much do you get paid for that? So the more time you take and the less you get paid, that's a good candidate for an AI, because an AI is likely going to be a lot cheaper than a human radiologist. And particularly applications that fall in this area that have high volume would be great candidates. And bone age, interpretation of bone age becomes, falls in that category. And I'll talk a little bit about that. The other way of thinking about this is how critical is the read. if a human radiologist can be highly accurate in making this read, then it's probably not that important. But let's say the error rate on the part of a human radiologist is pretty low. Sorry, the error rate is high, so the accuracy is low, and it's a pretty critical read. Then that is a good candidate for an AI because you can hopefully reduce some of those errors. So pediatric fractures, it turns out, fall into this category because the vast majority of pediatric fracture x-rays are actually read by adult radiologists who don't have subspecialty training, and they tend to make a lot of errors. So we've actually published on the error rate of adult radiologists reading pediatric fracture films. So that's another good candidate, and I'll talk to you a little bit about that. So the first project I'll talk about is our bone age work. And this was driven primarily by Nakul Reddy and Jesse Ryan. And if you're familiar with the process of interpreting bone age based on x-rays, it works kind of like this. You get a whole hand x-ray, and then you go to this book, and I'm not kidding, this is a real book. This is actually the one that's used in our department. It's the Grillick-Pyle Bone Age Index, or Atlas, Atlas for Skeletal Development. And why do you determine the bone age? It's important for a number of endocrine conditions, as well as preoperative planning for certain surgeries. This is actually the topic of the RSNA AI Challenge in 2017. So the way this works is you start comparing, you flip through the book and you compare features on the x-ray to features that are in the book, and then you read off an age on that basis. So the AI challenge that the RSNA mounted was great. A lot of companies, as well as a number of academic groups, came up with algorithms that were trained based on the database that the RSNA put out. And they came within a few weeks of the gold standard, which was pretty amazing. There have been companies that spun off those products, and they're used commercially today. But when we were doing this, we noticed, so my group also participated in this challenge. They actually came in, I think, number 6 in the challenge. And I think we were the highest ranked academic group. All the others were industrial groups with like huge compute capability. So when we were doing this, so one of the things you can do, obviously, is pick up a saliency from the neural network fit. And what we found was that there was a similar saliency in individual fingers as in the rest of the hand itself. And so we asked the question, can we just look at the index finger and get an accurate bone age? And why were we doing this? Well, you know, everyone's building algorithms for bone age, but what could we do different? And we were envisioning perhaps a little tabletop device modeled on a dental x-ray unit where, and this could actually sit in a endocrinologist's office or in a musculoskeletal surgeon's office, and they wouldn't even have to send the patient down to radiology in order to get a bone age. They just stick their finger into this device and it would read off a bone age. So with the kind of crazy dream in mind, we decided to see if we could do this with a single finger. And long story short, yes, you can. So what we did was we took the whole hand x-ray and then we trained RetinaNet to identify just the index finger. Well, the terminal phalanx, the intermediate phalanx, and a portion of the proximal phalanx. So it included 2 joints in the tip of the finger. And then we trained exception V3 on the whole hand as well as on single finger. And then we compared accuracy. So here are Bland-Alton plots looking at the bone age interpreted from the whole hand by the AI versus just a single digit. And as you can see here, they're roughly equivalent. You get about the same error. So the lesson we learned from this was AIs in this context can not only reproduce what a human reader can do, but they can probably do so with even less data. What I'm not showing you here was another extension of the study where we artificially noised this image. And you can actually have huge amounts of noise in this image to the point where it's completely uninterpretable by a human reader, and yet you get an accurate number out of the trained network. So that was a good learning lesson for us. The next project I'll talk about is the notion of making the AIs operate in ways similar to the way human radiologists operate. And so this was actually one of the first publications that looked at multiple views being coalesced into a single decision by an AI emulating a radiologist. And the particular context we looked at was elbow fractures, and replicating exactly what the human radiologist would do, look at 3 orthogonal views. So we trained for fractures using a consensus read of 3 certificate of added qualifications. So these are specifically pediatric musculoskeletal radiologists. So they're the best of the best as far as interpreting these x-rays go. Individually trained on each of these views, and then had a gating layer on top of that, which would basically assume that if we saw a fracture in any one of these views, then it would be counted as a fracture. And that's basically what we needed to get the accuracy of this algorithm up to the same level as human radiologists. And so here's a summary for supracondylar and lateral condylar fractures. The accuracy was pretty high, not so high in some of the other situations, but overall, we're pushing 95%, which was very good and comparable to what you can do with a trained pediatric musculoskeletal radiologist. The next project I'll talk to you about is looking at distal tibial fractures, toddler tibial fractures. And these are particularly important because toddler bones are continuously growing and they're open growth plates. And these show up in the x-ray as striations, which actually look a lot like fractures. If you're not a especially pediatric radiologist, you may not have seen a lot of these. It makes it difficult to interpret. So this was identified as a high need area. So we went ahead and collected a large number of radiographs in this period from our hospital, and they were reevaluated. Now, here's the interesting thing. In this 8-year period, we ended up finding about 800 cases. Well, maybe a little more, maybe 1,000 cases. Now, all of the previous networks that I talked to you about were trained on tens of thousands of cases. Now, yet, when you train a human radiologist, they definitely don't see tens of thousands of cases. And the question we were really asking was, is it possible to train a CNN, a neural network, perhaps using transfer learning, on a relatively small number of cases comparable to what a human radiologist would see during their training period? And the rationale, of course, is the same as it always is for transfer learning, in the sense that a radiologist in training is not naive. They know a lot about what's up, what's down, what's left, what's right. And so if you incorporate a previously trained to recognize images network and then operate in transfer learning mode, perhaps you can get away with a much smaller number of training set images. So that's exactly what we did. I'm not going to belabor the concept of transfer learning, but essentially we took ImageNet and we added modified top layers just to accomplish the fracture classification task. And you can see here the open growth plates that I was referring to, these striations, which could be very easily mistaken as fractures. And it gets really complicated when you have overlaid images like this. We also used a technique that's called N-fold training, where you take the same overall data set and you divide it up into a training set, a validation set, and a test set. And then you index these sets so that now you use a different subset of the overall set for training, validation, and test. And each of these folds, you can evaluate independently. You can get ROC curves for each of them, accuracy numbers. And you end up picking the best fold out of this as your selected network because it has weights that are tweaked appropriately to accurately classify the task. So we have this down to all incorporated into a single pipeline now, so it's relatively easy to do for any of our classification tasks. So the final system architecture for this system was you have individual views, and we had a YOLO network that was then trained to isolate the correct anatomy. And this is particularly important, as I'll show you in a second, because there's all kinds of complications in the images that need to be accounted for. And then you follow that with the fracture detection network that is trained on exception. And then, of course, you combine the multiple views with the gating layer that I told you about before to come up with a final decision. And you see here why it's really important to have this localizer to identify just the distal tibia. So this is where YOLO came in really, really useful. So this is actually output from our completed pipeline. The distal tibia is identified and it's labeled, and there's your final decision that's also been rendered. So this network, it turns out, this pipeline is incredibly accurate. We get numbers in the high 90s for this process. So the last topic that I'm going to talk to you about today is one that is particularly close to my heart. And this is the decision process for non-accidental trauma, which is a euphemism for child abuse. It's a terrible thing. Kids who are abused by adults who are normally tasked with their care. A lot of times it goes on for a long period of time before it's identified. So you have medical history for this patient. They see a number of different providers, but the notion of abuse hasn't really come up. And then finally, some kind of incident or occurrence that triggers the thought of maybe there may be child abuse going on here. And then whoever the physician who's seeing the patient at that time has a decision on their hands. Do you involve the child abuse team at that stage or not? Now, if you do, then one of the things that almost immediately happens is a call to social services. And it's entirely possible that the child will be separated from the adult at that stage. And if you're wrong and this is not abuse, then you remove the child from their parents' care or their guardian's care and got them into the foster care system. Now, on the other hand, if you don't make the call, then this patient might perhaps not get the care they want, and be sent back into an abusive situation. So it's a very weighty decision to involve the CAP team or not. And oftentimes the physicians who have to make that call are not trained in child abuse. So the number of times our child abuse team at Texas Children's gets phone calls from remote sites saying, I have this patient here, I'm not sure if it's abused or not, and can you help me out? This is a very, very common occurrence. So the idea was, can we take imaging data, lab data, as well as all of the medical history, and which is essentially the summation of information that this referring physician has to determine whether to refer to the child abuse team or not. And can we render a decision based on that with an AI? And that's exactly what we did. So we asked the question, how do you incorporate all of this data into one consensus decision? Because you've got numerical labs, you've got images, you've got medical history, and we finally decided that the best way to do this was to do natural language processing on the medical record itself. Don't look at the numerical values of the labs themselves, don't look at the images themselves, just look at the verbiage in the reports. And long story short, if you do that, you actually get a complete picture of the patient, and we get amazingly good accuracy. So using only the verbiage in the medical record prior to the first involvement of the abuse team, we can accurately predict child abuse in the mid-90% accuracies, which I thought was pretty amazing. This paper is in review right now, and hopefully will be out in the next month or so. So that's what I had for you today. Happy to take any questions.
Thank you very much for a great lecture. I don't see any questions coming up in the Brella chat window. We're a little bit over time, but I do have one question for you.
Sure.
You know, we hear a lot about big data sets in radiology for cancer, for brain imaging, but it seems like pediatric radiology is just different enough that those data sets may not cover that population. Are there big data pediatric radiology data sets that are out there or that are being built?
I can't tell you how many of them are being built or not. I know some are, but the notion of publicizing the data sets or making them publicly available, this is a topic of constant debate. I think people have realized that an algorithm is useful, but it's not as useful as it is after it is trained. And you can only train if you have high quality data. And therefore, the data is as important as the algorithm is to a potential final product. So the value of the data is highly recognized, and there's all kinds of jockeying going on to figure out who owns the data, who owns the trained algorithm. So
it's complicated.
I think it's going to be a while before this is resolved because,
Okay, well, very good. Thank you again for a great lecture. It is my pleasure to introduce our next speaker, Christy Brock, who received her bachelor's, master's, and PhD degrees in nuclear engineering and radiological sciences at the University of Michigan in Ann Arbor. She started her academic career in 2003 as an associate professor and physicist at the University Health Network in Toronto. She was certified by the American Board of Radiology in therapeutic medical physics in 2008. In 2012, she returned to the University of Michigan And then in 2016, we had the great fortune to be able to recruit her to the Department of Imaging Physics, where she's now serving as professor and executive director of our image-guided cancer therapy research program. Dr. Brock has held a number of leadership positions in the American Association of Physicists in Medicine, the American Society for Radiation Oncology, the Department of Imaging Physics. She's on the editorial board for the Journal of Medical Physics and the International Journal of Radiation Oncology, Biology, and Physics. Her academic focus is on biomechanical model-based deformable registration, which led to the development of an algorithm named Morpheus, which has been licensed by Research and incorporated into their radiation therapy treatment planning system. Her academic interests also include dose reconstruction to accurately estimate the dose delivered to normal and target tissues, and correlative pathology, correlative pathology. It's a tongue twister. And today she's going to speak with us about harnessing the power of AI for image-guided cancer therapy. Dr. Brock, the floor is yours.
Thank you, Dr. Bengtson. You can see my slides and hear me okay, hopefully. Excellent. So what I wanted to focus on today was applications of AI and image-guided cancer therapy demonstrated with liver ablation. So first, some disclosures. The work I'm going to present today is funded by an NIH Academic Industry Partnership grant in collaboration with Research, which has licensed my deformable registration algorithm. It's also funded by the Helen Black Image Guided Fund. And I definitely want to acknowledge that this is team science. Bruno Adisio is the PI of the clinical trial and my co-PI for the grant. And all of the AI work is really the brainchild of Brian Anderson, who is the extraordinary graduate student who is working on this project. I'd also like to acknowledge Kyle Jones and Gary Lynn, who have worked as well. So there's significant motivation for advanced image guidance for liver ablation. So this clock trial demonstrated that chemo plus radiofrequency ablation outperforms in survival chemotherapy alone. We also know from the study on the right that having sufficient margins around the tumor is critically important to progression-free survival. So these combined together makes us look at the image guidance process for the ablation. So despite advances in imaging, targeting accuracy and ablation margin coverage is still a source of uncertainty due to the lack of tools to integrate the images. So why is that so challenging? And it's really because of the complexities associated with the liver. So you can see on the left here, this is a contrast enhanced CT scan. So you can see the vasculature and you can nicely see the tumor. So we can contour manually the tumor here. However, then when we go into the treatment room, we can get this at the start of treatment, but then the radiologist, the interventional radiologist will look at this and determine what the trajectory is to insert the ablation probe. They then acquire another CT image with this probe in place, but you can see that the contrast is washed out, so you can no longer see the tumor. As well, if you did administer additional contrast, then you wouldn't be able to see the tumor very well because of the artifact that is resulting from the ablation probe. So we must be able to integrate this information together to be able to map this tumor onto this ablation region. In addition, immediately post-treatment, we can acquire another contrast enhanced scan and you can then see the ablation zone. However, the ablation zone and the tumor show up identical on these contrast enhanced scans. And so the question still remains exactly how much coverage is provided by this ablation margin to the tumor because of the deformation that occurs. And so the idea is if at this point we could map the tumor onto the ablation region, then before the patient leaves the ablation suite, we could determine whether additional ablation was needed to ensure that we had sufficient coverage, leading to that higher chance of local control. Now, deformable registration for complex geometry and variable intensities, as we see here, is very challenging. The majority of deformable registration algorithms use intensity to drive the registration. So they look at the variation in the contrast on this image and try to match it to this image. So this causes significant challenges when your secondary image lacks contrast, has significant deformation, and has this change in intensity. So the goal is not to align this dark tumor region exactly to match this dark ablation region because they represent different geometries. Now, biomechanical model-based approaches may provide a solution to this. And so my lab, as Dr. Bengtson had mentioned, has developed Morpheus, which is a biomechanical model-based deformable registration. So simply put, it takes the liver contours on one scan and converts it to a mesh, as you see here. It creates a surface mesh and then a tetrahedral mesh covering the entire volume. You can add into that the tumor and the vasculature if you'd like, and then you can assign different material properties to these different structures. Between the two images where you have the surface meshes, you then find a correspondence that serves as the boundary conditions. And then when you put these into a finite element analysis, it solves the deformation and moves the tumor and deforms the tumor according to the biomechanical properties. So that means it's moving the tumor and mapping it onto the secondary image based on the linear elasticity of the model. So the stiffness and the compressibility, rather than trying to align the intensities. So this enables mapping of the tumor onto a secondary image with the same level of accuracy if you have two beautiful diagnostic quality contrast enhanced images, or if you have a secondary image with less contrast. We validated this in numerous liver cases and found it to be on the order of accuracy on the voxel size. However, it does require segmentation. So you need to have a liver segmentation on both images, and it can take approximately 2 to 4 minutes to run. So the question we had was, if we take these kind of tools, could it potentially make a difference in outcomes for liver ablation cases? So we did a retrospective study where we looked for a fairly even subset of patients that were previously treated and found 14 with a local recurrence and 16 that had significant follow-up with no demonstration of local recurrence. We selected patients that had a lesion size of less than or equal to 3 centimeters and that they had contrast enhanced CT images within 45 days of treatment and a contrast enhanced CT image immediately after ablation. So what we did is run the biomechanical model-based deformable registration to map the pre-treatment imaging tumor onto the post-treatment image and then measure the ablation margin. And then we looked at what the difference was between the cases that had a local recurrence and not a local recurrence. So this plot shows for the local recurrence and the non-local recurrence cases, the minimum margin between the mapped tumor and the ablation zone. So you can see a case where you have basically complete overlap between the tumor edge and the ablation zone versus a case where you have sufficient coverage. And you can see the dramatic difference between the local recurrence cases and the non-local recurrence cases. And this was statistically significant using a Mann-Whitney U test. So this gave us a lot of motivation to try to translate this into a clinical trial. So given this highly significant correlation, is it possible to translate this technology into the treatment room and be able to evaluate the probe placement and the ablation margin in real time? However, in order to do this, we need to be able to automatically segment the liver and the tumor and the ablation, as well as do efficient deformable registration. So when you're manually contouring the liver, it takes on average about 20 minutes, which is makes it completely prohibitive for the interventional suite. So of course, insert AI. And so Brian started investigating several different deep learning methods. And so those are shown here. I'm not going to go into all of the details. What we found was that the Deep Lab V3 Plus algorithm performed the best. And I'll show you the quantitative evaluation here. So we also wanted to make sure that this model worked on multi-institutional data. So we found patients from MD Anderson, which are shown here, as well as a MICAI abdomen challenge. We also wanted to make sure that the algorithm worked on contrast enhanced images as well as non-contrast enhanced images, because we know that the contrast level varies and that we would have both. So we wanted one algorithm that would work on all of them. We split this data into training, testing, and validation as shown here. And here's the result. So this is a Deep Lab V3 Plus model with an exception backbone. You can see the data sets with the disease with contrast, the non-contrast, and then some normal images. So these show the patient number. This is the externally withheld validation set, as well as the dice similarity coefficient, which measures the overlap between the AI segmented liver and the manually segmented liver, as well as the mean surface distance. So you can see that the performance was consistent across all of these different image sets and was very high and well within the inter-observer variability, and the mean surface distance was very low. We also wanted to make sure that when we looked at this qualitatively, it also held up. So we know that sometimes with a big organ like the liver, errors in the contouring can be disguised by high DICE values, even when there are significant errors. So we looked at a cohort of patients that was treated in the radiation oncology department here at MD Anderson and found 50 patients, 25 that were HCC and 25 that had liver metastases. So the clinicians, we had 3 clinicians, 1 clinician performed the evaluation twice and 2 others performed it once. And we did a blinded comparison between the previously manually defined clinical contours that were approved and used for treatment versus the AI-based segmentation. So the top here shows the majority voting. So that was the majority of these 4 votes. And you can see that the preference was for the AI-based segmentation of 60% compared to 40%. And you can see the clinical usability. And then this shows at least 1 vote. And you can see that in 48 out of the 50 cases, so 96% of the cases, at least one of the users felt that the AI-based segmentation was clinically usable. So we felt like this was a very positive result and that we could use this to translate into our clinical trial to guide the focal liver ablation. Now, contouring and identifying the tumor and the ablation region is much more challenging. And so Brian developed this very elegant hybrid WNet. So there's a pre-trained 2D dense net that's converted into a UNet where the features are concatenated into a 3D dense net. So you can see the complex nature here. Again, this was developed and trained on multi-institutional data. So we took advantage of some challenge data that was available. You can see the division between the training and the validation, as well as the 2 test sets that were here. So you can see how much more challenging this is to be able to identify this disease, as well as the variation in the disease size and the variation in the contrast that was present in these cases. So this demonstrates the quantitative result. And so for this 3D IR CAD data, Brian was able to evaluate this on tumors that were less than 15 millimeters and then greater than 15 millimeters. And this is the DICE similarity coefficient. So you can see that we have a significant drop off in the DICE for the very small tumors, and the sensitivity of detecting those decreases. However, once you get above a diameter of 15 millimeters, which is the size of most of the tumors that we're treating in the liver ablation, the DICE similarity coefficient is quite high and the sensitivity is quite remarkable. When we evaluate that on the total cohort, you can see the LITS challenge, the 3D IR-CAD, as well as the MD Anderson colorectal METs. And then we use this algorithm to segment the MD Anderson liver ablation. So again, you can see very high mean DICE similarity coefficients. Again, that ranges from 0 to 1. And when we look at inter-observer contour variation, we typically see around 0.7 to 0.8. And so the difference between a single observer and the AI is on that same order. We also did a qualitative evaluation at MD Anderson, and you can see that here. So we used this Likert score ranging from 1 of very poor to 5 of excellent, and evaluated that on 3 independent reviewers. So you can see for the colorectal liver metastases, looking at 24 cases, all of the cases for all of the reviewers were either a 4 or a 5, ranging from good to excellent. And then when looking at the ablation region, you can see that there were a couple of cases that had poor or fair, but the vast majority were scored as good or excellent. So again, being very excited that this led to encouraging results that we could use in the treatment room to guide the liver ablation, because we could always have the opportunity to edit the volumes if there was the need at that time, but that this would give us an excellent starting point. Now, I also mentioned that the deformable registration can take a couple of minutes. And so this is still somewhat of a work in progress, but we wondered if we could use a deep learning UNet with Atrius convolutions to be able to predict the biomechanical registration. So we took the deep learning based segmentation, fed it into the Morpheus deformable registration, and then use this to train the UNet. So we're still in the process of running some data through. We have 58 patients right now, 50 for the training and then 8 for the testing. And you can see the very encouraging results that are here. So we evaluated this using spherical coordinates. So 75% of the points in the liver were within 1 voxel value. If you look down here, this is the GTV centroid difference in the 16 cases that were mapped. And you can see that this is all within the voxel size that has a slice thickness of 2 millimeters. And then this is the results of all of the points within the liver, each of the voxels in terms of magnitude and then the direction. And you can see the magnitude difference here with 75% within the voxel size. So again, very encouraging results. And we're using both the AI segmentation of the liver, the tumor, and the ablation for our NIH-funded randomized phase 2 clinical trial, which is shown here. We're not using the AI-trained biomechanical model. We are spending the 2 to 3 minutes to do that deformable registration at this point to make sure that we have the highest level of accuracy. But we're using this to segment the tumor, to deform the registration, do the deformable registration, and then both confirm the needle position as well as to estimate the ablation margin. So the last thing that I would like to touch on is very much a work in progress, but is it possible, given all of this data, that we can just use AI to predict the recurrence risk? And so this is what we're working on now in collaboration with a collaborator in Brazil, so that again, we have the multi-institutional data. So the idea is if we put into the network the pre-treatment liver and tumor, as well as the post-treatment liver and ablation region, as well as the clinical factors that we think are important, and we put this into a residual block with some dense nets, can we predict whether the patient will have a recurrence or not? And that's a work in progress. So I don't have any results for you yet, but we're looking forward to seeing what kind of level of accuracy we can have there. So in conclusion, image-guided liver ablation has been shown to improve overall survival, and we may be able to further improve this with advanced image guidance capabilities. Liver deformation, probe insertion, and image contrast make this registration challenging. However, we believe that this biomechanical approach can overcome this. However, it requires segmentation and a couple of minutes in the room. AI has shown tremendous potential to overcome these efficiency limitations and has allowed us to begin this phase 2 clinical trial, which we're very excited about. And the impact of local control and survival will come out in the next few years. So we're looking forward to that. Thank you for the opportunity to speak with you today.
Well, thank you very much for yet another great lecture. We have time. We have a couple minutes for questions. I don't know. It looks like we may have a lot of attendees with broken fingers who can't type questions into the chat box umbrella. Please do so if you have anything. Don't be shy. I have a question to kick off the conversation, though. So I have this perception that there's just a huge amount of pain in terms of clinical translation of algorithms and updates to algorithms on clinical devices, right? So has adoption of updates to Morpheus been problematic on clinical devices? And do you foresee that there will be just a high barrier for integrating AI as a supportive tool in this area?
Yeah, so that's a great question, Jim. So the infrastructure that we use in RayStation is nice because it has this scripting environment. And so the AI-based segmentation is scripted into it. So we're running that on our platform. So it doesn't require kind of complete integration into their commercial software package. I mentioned the amount of time that it takes to do the deformable registration. So we did work with them. We have a strategic alliance with them, and as well as this academic industry partnership grant. So we did work with them to try to speed up and to increase the efficiency of the Morpheus as it is implemented in their algorithm. And we were able to decrease it from 6 minutes down to that 2 to 3 minutes. So we're really excited about that. And we saw that translation happen very quickly.
Are you thinking that the whole biomechanical deformable registration process algorithm can become AI rather than
Yeah.
the analytical solution.
I mean, that's really the interesting thing. And, you know, the question really remains as to how many cases do you need to run it on to have that level of confidence? Because the problem with this is, you know, the benefit of the biomechanics is that it allows you to do deformation with the same level of accuracy that you validated when you can confirm the accuracy. And so in these cases, you're doing the deformation and there's no way to measure the accuracy of mapping that tumor because you can't see the tumor. There's no metrics to know whether it's right or not. And so you rely on that fundamental understanding of the biomechanics. And so replacing that fundamental understanding of the biomechanics with an AI approximation makes me more nervous. And so we'd have to do a lot of validation and testing to make sure that this wasn't an outlier case that was going to fail and give us, you know, erroneous results, because those in this situation would have profound impact for the patient.
Right. Yeah. And then last question, how are you training your outstanding students to be able to work with these and develop deep learning algorithms? What's your approach?
Brian is a total rock star. So he has taught me way more about AI than I have ever taught him. And so, you know, it's great because he learned a lot from taking the RICE class by Dr. Ankit Patel. So we're very fortunate to have a close collaboration with the RICE team.
Great. Thank you very much. Fantastic lecture. I appreciate you taking the time to speak to us today. Next up is Dr. Rifat Gaber, who is an associate professor in the Department of Radiology at UT Health and the McGovern Medical School. He received his bachelor's and master's degrees in biomedical engineering from Cairo University, and his PhD degree in electrical and computer engineering from Johns Hopkins. In his postdoctoral and doctoral research, he developed techniques to quantify heart muscle energetics in human heart failure using MRI and phosphorus MR spectroscopy. His current research interests are in quantitative MRI, patient adaptive MRI, myelin imaging, advanced image analysis, deep learning, and the applications in multiple sclerosis, Huntington's disease, and stroke. And today he's going to be presenting on the effects of training size on deep learning-based neural tissue segmentation of MRI in multiple sclerosis. Another tongue twister, at least for me. Dr. Gaber, the floor is yours.
Okay, thanks, Jim, for the introduction, and thanks for inviting us to present on this topic. Dr. Narayana was supposed to present this talk, but unfortunately, he's not able to join us today. So I'm going to present. So this is a study that we did to investigate the required really size of data that's needed for the segmentation of brain images in MRI. So deep learning, as we know, just like it has great promise for image segmentation in various medical imaging fields, and multiple sclerosis is no exception. So it has been very active area of research. And the advantage of using deep learning over conventional machine learning technique is that you don't need to handcraft image features. So you can just let the deep learning model learn from the data itself and discover the relevant features. And one of the most successful deep learning model is the UNet, which is widely used. It's a fully convolutional neural network. And it has two paths, like encoding really features and then decoding these with the skip connections to preserve resolution. One disadvantage of deep learning is that it requires a large amount of data to train. And unfortunately, it's very hard to actually come to annotated data in medical images, because this is a very expensive task to perform, to go over a large amount of images and then label actually the different tissue classes. So so there are a few techniques that try to actually to reduce the required training size. And the most widely used of this is transfer learning. We actually take a model that's developed somewhere else and then fine tune it so that it works better for your task. And it will be better if the network is trained in a related field or a close field rather than just a generic computer vision type of data. And the third technique actually to reduce the image sizes, to reduce the training sizes, to do data augmentation, to artificially create data or variations of your data to use into the training. But for now, like to really to train an image or to train a DL model, you still need a large amount of data. And you actually want the smallest possible amount of data to train, because the data is not always available. But it's not really, when we started doing this in multiple sclerosis, we didn't know really what's the minimum size of the data. And the more we did research, we didn't actually find really an answer. Really, nobody did this, even though there was some investigations on general medical imaging using a smaller data size. There was no systematic study that looked at the training size and its effect on the segmentation accuracy in MS. And we are fortunate that we have a large data set that actually helped us to investigate this question. So, and the tool that we use for the study is what they call the learning curve, which is an equation that describes how fast a machine learning or deep learning model learns from the data. So it's described by this equation, which is more like an exponential kind of recovery in terms of MRI notations, and it really describes how fast the model learns. So so we wanted to exploit the large data set to actually see how our model learns. So the data that we have in our lab is, it comes from a clinical trial called the COMBI-RX trial, which is a multiple sclerosis trial of a combination therapy for MS. And it has more than 1,000 patients that were scanned at baseline, coming from a variety of 68 centers actually that participated, most of them at 1.5 Tesla and some at 3 Tesla. And the MRI protocol that was used in this study, it acquires 2D FLIR images, 2D dual echo, turbospan echo, and pre and post contrast T1 weighted images. All of these are like 2D images, so this is kind of the scan resolution, 3mm slide thickness. So all of these images went through standard kind of pre-processing steps to really get to the brain images. So you start by skull stripping, you do bias field correction, intensity normalization or standardization, because MRI really doesn't have units. And also we do some denoising of the data. The ground truth for the segmentation came also from the COMBI-RX study, and that was actually kind of come from a semi-automated software, the MRI AP, which is also was developed in our lab. And after the segmentation of MS, the data were validated and reviewed by actually MS experts who actually delineated the tissues. So that constituted the ground truth for training. So this is kind of an example of the data. So the protein density, T2, FLIR images, pre-T1, and also have post-T1, even though we didn't use it in this study for the segmentation. We use the standard UNet. So as I said, it has like two contracting and expanding paths with a skip connection. Very, very powerful and very popular architecture for image segmentation. So now for the network training. So the inputs to the network were actually the 4 image sets that I just mentioned, and the validated segmentation used as a ground truth. And so the 1,000 images kind of remaining after execution of 8 data that really had the artifacts, they were partitioned into training, validation, and test set with an 80%, 10%, and 10%. And further, this 80% was actually kind of sampled in different ways. So we could use the whole 800 images in the training set for training, and now and then we move to 500, 400, 250, and even down to 10 images, image volumes for the training. And then for each of these, we trained the network, and we evaluated the dice similarity coefficients, how good is the segmentations for the different kind of tissues in the brain. And also we fitted the dice coefficients to this learning curve and determined the parameters and also the confidence intervals for these parameters. And to see how good is the model fit, we actually used 3 different data sizes, training sizes, 50, 70, and 90, to train the model. And then we predicted the dice coefficients for larger data set. And since we actually have the data, so we can actually validate how good is this prediction. The last kind of evaluation that we did is to look at the effect of the lesion size. So you expect that the smaller the lesion, the harder it is, so the more difficult to segment. And also we computed, in addition to the dice, we computed the true positive and false positive rates for these lesions. So these are like computed lesion-wise rather than voxel-wise. Some results. So this is kind of the input flare image. And at the bottom here, you see the ground truth for the segmentation. So CSF is in cyan color, and then gray matter in gray, white matter in white, and then this salmon color is the MS lesions, or the lesions detected in this image. So A, B, C, D through F, this is the different training sizes that were used. So A is using 800 data sets, then using 400 images for the training, then 100, 50, 25, and 10. So, you know, it's like there's reasonable performance until actually the training size fell to 25 or less. Now you actually completely miss the lesions, which is kind of segmented as gray matter. So that gives you like really what's the training size that you need for this image data set. So this is kind of the evaluation of the network output on the test data of 100 cases, and we're just plotting the volume of the different tissues in the ground truth against that in the from the UNet. And this is for all the tissues and the lesions. And actually, when you use all the data, it's excellent agreement between the two. The effect of the training size also, when we fitted the learning curve, this is kind of the exponential curve, it fits very, very well with the data. So this indicates that actually the learning curve is a very good fit to the data, and we can use it in practice. And this is the prediction. So we use like images with the small data sizes up to actually 9 subsets, or actually 90 cases for the training. And then we predicted what will be the dice coefficients using larger amounts of data. So the data that's used here for the fit of the curve is the black circles, and the white circles, or the light circles, is the predicted. And it falls nicely actually in the middle of the confidence interval, which means it's really a good model. Looking at the lesions, so this is the different number of the data sizes. So the small data size of n equal 10, usually it's very difficult. You cannot really detect lesions with 10 or 25. 30 start to really improve much, like the significant improvement at 30. But at 50 really is like really you get excellent performance. So this is kind of what we think is the best choice for segmenting MS lesions. Looking at the data, the lesion size, of course, so the lighter color here represents a smaller lesion size. As the lesion size increase, you consistently like improve your performance. So this is kind of as expected, and is also reflected in the true positive, which improves with the larger lesions. and also with the false positive rate, which also consistently decrease. So overall, actually, this is very good performance of the learning curve, and the availability of a large data set like the compare X. This is really what helps you to answer these kind of questions, which is really if you have 100 data set and you train a model using 60 set and test in 40, you don't really know if 60 is sufficient or not, or if this is the best or not. So this is really this is where big data really helps to answer these questions. And the learning curve really here has shown good predictive value. And the network overall, it produces very good correlations with actually the ground truth. So we would recommend that using actually 50 data set or so is the minimum that you'll need for MS cases. Um, but actually for other brain tissues like gray matter, white matter, and CSF, the performance was really very good even with data set as small as 10. And one reason for this is that these tissues have a larger number of larger volume, larger number of voxels to train, while actually the lesions are the smaller kind of smaller fraction of the volume. So it's more hard to segment. Uh, um, so looking at the lesion size, we can actually get an accuracy of about 80% for lesions as small as 500 microliters. So this is pretty good actually for MS. Um, and while actually it's very difficult for very small lesions, even if you use a training size of 800, which is the largest, it's very hard to really uh, 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 to get lesions smaller than 70. So finally, I'll just like mention few limitations about this study. So the training size, of course, depends on many factors. One of them is the acquisition protocol that we're using. So we used 4 different image contrast: flare, proton density, T2, and T1. So the question is like, okay, how much of this you need, or how if you just use flare, how much is this going to affect the training size? And this is something we're investigating also on our lab. So we recently did a study actually comparing the different pulse sequences uh, and their performance on the segmentation. Um, it would be nice really to combine the 2 studies and see the effect of the training size in each of these. Also, the network configuration, the network architecture can also affect this study, affect the results. So there's kind of some caution, like when we want to generalize these. But our feeling is that this is really actually, it gives some at least initial value that you want to consider when you're actually designing your deep learning classifier. And finally, like if you want to really learn more details about this study or the results, please see our recent publication on the topic. Thank you.
Thank you very much, Rafat. Great lecture. We have time for a couple of questions from the audience. And in the meantime, I've got a couple that I'd like to ask you. So it seems like this is an important approach to lots of different kinds of applications. So you found that a training set of 25 to 50 is sufficient for your problem. Do you think that that's and I know that simpler tasks will need fewer, and more complicated tasks will require more. But is that a reasonable ballpark for most applications, or is the range going to be huge of training data sets that you'll need to train a model?
It's really difficult to answer this question like for all applications, since applications really vary widely in the degree of difficulty. But my feeling is that for MS segmentation, this is pretty much kind of reasonable number, and we didn't notice large variations when we were playing with different DL models. They were kind of also kind of similar. So maybe the model didn't affect things much. So it looks like 25 to 50 is the minimum that you need for MS lesion segmentation. And as I mentioned, like just like for brain tissue segmentation, if you want just to do gray matter, white matter, CSF segmentations, just 10 cases were actually sufficient for that purpose in our data. So of course, I mean, the difficult the target, the more the training data that you need.
Wonder if have you thought about ways to create a prospective tool for power analysis? I'm not sure how you would capture the complexity of the task that you're training for, but you have this learning model, and I wonder if those parameters could be sort of heuristically set. Okay, this is a really complicated task, therefore this parameter becomes high, and how does that guide us? It
Yes.
seems challenging in that you can't really do a power analysis until you're significantly underway on the problem already.
Yes, we kind of published the fitted model for this, the fitted learning curve. So this is we think that that could be used at least in similar studies to predict the anticipated accuracy, really. Um, Now it's generalization is always a difficult problem really in deep learning. So it remains to see really if say if another group will say, okay, no, your equations doesn't work for us, or it actually works very well. So this is something to look for.
And we have a question from the audience. I'm so excited. What optimizer did you use? Did you see any differences in the optimizer convergence between the larger and smaller data sets?
Uh, I I think we used Adam for optimization. And like in my experience, like we really see differences in the performance of these. Like uh, when we use like SGD or Adam or different ones, we see some sensitivity to these. Like it's kind of like it's more of an art, I think, because it really varies in unexpected ways. Um, so so that, I mean, we use Adam, but I'm not sure that's the best thing to use. And there are different variations on these still coming. So it's always kind of a changing kind of tool. I'm not sure that answered the question, but
I think so. One last question for you. So in image reconstruction, often people will use these bootstrapping methods, you know, where you may just use the same or very similar data, but add different amounts of noise, or swap some of the encoded data, swap, you know, take two sets of images and then various permutations of the case-based data in order to generate a lot more training data.
yes.
How widely is that approach used in training of machine learning algorithms?
I think it's quite widely used. We use it also in some applications when where we don't have enough data really to split things easily into training and test, as we did in this study. But the one technique that's many people use is cause validation, which is basically kind of it's kind of similar to leave one out or leave one sample out, but on a groups really. So you put your data in buckets and just you leave one bucket out, and then you cycle the assignment of the testing bucket and evaluate. So it's very similar really to bootstrapping in
Right.
that sense.
Okay. All right, any more questions from the audience? Silence. Okay, if not, then thank you very much, Rafat, for a great lecture. And now let's see,
Thank you.
we're up to the next segment of the meeting today, the poster data blitz, which is always a ton of fun. So here's what's going to happen over the next little while. Each of the trainees who have submitted a poster are going to get 1 minute to give a little power pitch and compel you to attend their poster session. And then once those presentations are done, Don Koob is going to give us a quick rundown on how you can visit each of these posters individually to chat with the presenters about their work. And then we'll have lunch, networking, and poster presentations. We'll have about an hour and a half for everybody to take a break, eat lunch, and cycle through the various posters. So let me get back to the schedule. Our first presenter today is Brian Antonio. My old eyes are too small are too bad to see the text of your institution on the screen.
I'm coming from Rice University.
Brian Antonio from Rice University. There we go.
Yeah,
all right,
thank you.
Brian, take it away.
Thank you for the introduction. So in this work, we used machine learning algorithms to decode both signals associated with tongue motor control. And the relevant data here was acquired via functional MRI experiments in which healthy subjects were instructed to position their tongue in a specified direction. In some cases, the subjects also received a neurofeedback stimulus based on how consistently they performed the task. We tested the performance of three machine learning classifiers, which include linear SVM, radial basis function SVM, as well as an artificial neural network. And these algorithms were evaluated on two specific classification tasks. First, we used the classifiers to decode whether the subjects had actually moved their tongue. And second, the classifiers were also used to detect whether the subjects had actually received a neurofeedback stimulus. And on average, we found that these classifiers performed with prediction accuracies of at least 70%. Thank you.
Great, 50 seconds. That was perfect.
Thank you so much.
All right, next poster, please. Next is Nicholas Fulerchek. From Baylor. Is that right? Nicholas, you on?
I don't believe he's on.
Okay, then we'll move on to R. Hekmati.
Yes, thank you.
Are you here?
Yep.
Rasul, okay, also from Baylor. The floor is yours.
Thank you very much. In this study, we quantified the bold neuromodulation generated by neurofeedback for cranial nerve injury rehabilitation. The goal is to enhance consistency of voluntary tongue movement in 4 directions in healthy subjects. Then it can be applied as a therapeutic modality to patients with this kind of injury. Tongue movement is accomplished by cranial nerves, and any damage to these nerves can result in oral pain, swallowing, mastication, and speech impediment. Our hypothesis is that area under the curve generated by BOLD is increased and variance of BOLD is decreased. It's between 10 and 20 and 60% for area under the curve and at least 20% for variance. We use techniques like D primes as effect size between control and neurofeedback distributions, and methods like TSNE to visualize data in lower dimensions. The study suggests that the induction of neurofeedback can achieve enhanced control of tongue movement. Thank you very much.
Perfect. Thank you very much. Next up is Dong Huynh from Baylor College of Medicine.
Hi, everyone. I'll be presenting a brain-computer interface system using real-time fMRI neurofeedback our lab has developed with the goal to apply it as a therapeutic modality for patients with impaired tongue movement following lower cranial nerve injuries. Our study examines the effectiveness of our method on healthy subjects. Using support vector machine, we show that neurofeedback can enhance consistency of brain patterns associated with the control of tongue movement. Neurofeedback also increases the magnitude of both signal in motor, sensory, and attention regions by 34 to 52%, but no effect was observed for reward regions when compared with the control condition. These results suggest that neurofeedback can serve as a neurorehabilitative method for patients. Thank you.
Great, thank you very much. Next up is Rahul Kadakia from UT Health or University of Texas.
Yeah, UT Austin. Thanks for the introduction.
Welcome. The floor is yours.
Thanks. Yeah, so my lab and my research predominantly focuses on using fluorine MRI for diagnostic. I've worked on a couple of different projects in the past, but this poster focuses on detecting cellular hypoxia. Many people may or may not know this, but copper ATSM is a well-known radioactive drug that's selectively uptake by hypoxic cells. What we in the K-Lab have done is essentially fluorinated that probe and then made a second and third generation of that, where we've added a fluorophore for not only 19F diagnostics, but also we can use a fluorophore for cellular localization, as well as increase the amount of fluorines per probe to have just better or decreased ingestion concentration, as well as just signal intensity. So we've formulated that into nanoemulsion, and then real quick, ongoing work focuses on using different types of appended fluorine atoms. So CF3 bonds is what we used in the past. Now I'm focused on using SF5 bonds. So if you're interested, come on over to my poster. Thanks.
Fantastic. Thank you very much. Next up, V. Lee from UT Health.
Yeah, thank you. So in my project working with McConville Medical School, I'm working on a project focusing on MRI for multiple sclerosis patients. So in that those MRI, we see a fuzzy border area of subtly increase in the intensity, and the manual segmentation of this also caused like diffusely abnormal white matter or DOM. So the manual segmentation of DOM proved to be challenging and time consuming. So in this project, we propose a framework using UNet to automate the process of segmentation of DOM from all the components of the brain component in MRI. And even though this is still an early progress and still work in progress, we have some promising results to as a foundation for future implementation of UNet. So come join my presentation in person later.
Great. Thank you very much. Up next is Rob Raphael from Rice University.
Hello, I'm Rob's graduate student. I'll be presenting this short talk. And good morning. What we'd like to really share quickly are preliminary images of the mouse vestibular system, which we've taken using optical coherence tomography. And for the first time, we're able to image and see intact membranes within the bony semicircular canals. And this is particularly exciting because it lets us investigate disease conditions such as Meniere's disease, and which involves changes in these fluid compartments. And today, we're particularly interested in how we can use image analysis and other techniques discussed to analyze our data and images going forward. I'd love to talk to you more about this, and please drop by my poster today. Thank you.
All right, great job. Last but not least, Ninchao Wang from Texas A&M University. You're muted. You're muted. There we go.
Okay, so hi everyone. Our project focuses on developing image processing methods for automated cell segmentation and also machine learning algorithm for functional classification of cells within autofluorescence lifetime images. So for cell segmentation part, after pre-processing, we managed to get the clean cell masks, and then a watershed algorithm was used to solve the cell cluster problem. After that, we use multiple features to filter out the noises. As for the functional classification of cells, we adopted multiple machine learning methods to identify drug-resistant tumor cells and T cells within tumor masses. So if you're interested, please join my Zoom link. Thank you.
Great. All poster presenters did a fantastic job with their power pitches. Thank you very much. Now, Don, can you take us through how this is going to work? This is the first time for me to use Brello as well. So if nobody else needs the lesson, I do.
Okay, can everybody see the Brello platform on my screen? Perfect.
Yes.
So I'm in the schedule right now. I'm going to click on poster presentations. You'll see the title of the poster and the presenter. You click on the first one. This is their poster, contact information. We'd like to read the abstract. You can chat with them, and then at 12:10, everyone will be in a Zoom, their own Zoom room, where you can click on here. It'll open a Zoom link for their presentations. Anybody have any questions?
Fantastic. Are there questions on this? I would recommend that not everybody start at the first speaker, because in that list view, if everybody starts at the top and works their way down, then there will be 20 people speaking to the first person and nobody speaking to the person who's last on that list. So please pick a random point. Some start in the middle, some start at the bottom, and let's go visit these posters and learn from our future imaging scientists. And then we'll reconvene at what time are we getting started again? At 1:20 for the keynote lecture from Christian Badea.
Okay, I want to welcome everybody back. Very sorry we had a few technical glitches. The reason we're starting late, so we will probably go a little bit into the next break, but we'll go ahead and get started. Briefly, I want to mention that if you left your Brella tab open during lunch, you need to refresh your browser. Once you've done that, you'll notice on the left-hand side, there is a stream tab. All of the talks will be streamed there, and you'll notice on the right-hand side of that, there is a chat. And that'll be the same chat for all speakers this afternoon for the questions for you to enter so that we can get those to the speaker. We had some confusion earlier, so we just want to make sure that everybody's on the same page. Again, the stream tab on the left-hand side of your computer screen, that's the page where you want to be to enter questions into the chat for the speaker. Also, if you want to see any of the past content to see if questions were answered earlier from your schedule tab at the top right, you can click a box that says past content, and that will allow you to look behind to see if any of those questions, see how the speakers responded to the questions that were entered into the chat. So now, without further ado, it is my privilege to introduce Dr. Christian Badea, our next keynote speaker. He's a professor in the Department of Radiology and faculty in departments of biomedical engineering and medical physics at Duke University. His research interests are in the physics and biomedical applications of computed tomography, micro-CT, tomosynthesis, and image reconstruction algorithms. Currently, he's the co-director of the Quantitative Imaging and Analysis Lab with a mission to develop, optimize, and apply novel CT and MRI quantitative imaging at both preclinical and clinical levels. And today he's going to talk to us about deep learning approaches in spectral CT. Christian?
Thank you, Susan. You can hear me?
Yes, sounds good, looks good.
Thank you for being here. We had some technical glitches, but without further ado, let's start this talk here. And I was very pleased to see the talks this morning. They were very different, and this talk, it's also different. I'm a tool builder, and we're going to talk about spectral CT, which I believe is the future of CT imaging. challenges associated with spectral CT, as well as some solutions that we have provided to these challenges through deep learning. An introduction, contents of my talk are shown here, because I assume not everybody is an expert in spectral CT. I'm gonna spend some slides trying to present the major concepts in spectral CT. I'm going to show you a number of preclinical prototypes for spectral CT that we have developed at Duke. And then I will focus for some of the imaging challenges. Again, we will stay in the field of image formation, image reconstruction, and problems that we faced there when we build some of these prototypes, and then how we have used deep learning approaches to address some of these challenges. There will be three case studies, if you like, or three applications of deep learning for preclinical CT. And I will end with a clinical CT, spectral CT, dual source CT application of deep learning. So that will be a clinical application. So let's start with basic concepts for CT imaging. If you go to the hospital, the majority, excuse me, of CT systems will be single energy CT. What it means is that we have an X-ray source, we have a detector, which we call a black and white detector, and the source and the detector, they are mounted in a gantry that rotates around the patient. And in essence, we measure in CT absorption of the X-rays, and so it's linear attenuation coefficients that we try to image with CT. Now, if we normalize those measurements to water, we can create these CT numbers or Hounsfield units. But in essence, you know, with a single energy CT, and by single energy, we mean a single measurement, because in reality, we have an X-ray source, which is polychromatic. So it's a distribution of X-ray photons, but the detector in this case is integrating all the information for each voxel in a single number, that CT number of Hounsfield units. There are many things you can do, but not enough, let's say, especially in terms of contrast discrimination. CT is not very flexible like MR it is in trying to manipulate your sampling sequences or something like that to be able to get different contrasts. Well, there is something there. There is a dual energy CT. Again, this is something that exists in the clinics maybe for 10 years now. And there we have different manufacturers that have different solutions, but in essence, you have two measurements. And what I see here is that, excuse me, with two black and white or grayscale detectors like that, we can essentially sample those linear attenuation curves that we show here in 2 points with those two measurements. And that's helpful because you can differentiate between, for example, iodine-based contrast agents and bone, which would be calcium-based, or soft tissue. So it gives a bit more contrast discrimination that way. However, that's not enough. And I believe the future of CT is in using photon counting technologies. There are new X-ray detectors that they have appeared the last couple of years. These X-ray detectors, I'm simplifying here, but they act more or less like when you have for light a prism, you would separate the white light in different colors. Again, this is more of a metaphor, but in essence, the polychromatic distribution of the X-ray source is kind of separated into different energy beams. So we can have ways to sample those linear attenuation curves in multiple measurements like that. And as expected, we are getting more discrimination, contrast discrimination, so that the promise is that we can use multiple contrast agents that will be tagged with different materials, having different K edges, that will be able to separate simultaneously in the body. Okay, so if you look on the right side here, both the single energy CT and the dual energy CT, they are based on this black and white, I would say, or energy integrating detectors. On the other side, you can see that we refer to spectral CT to both dual energy CT using energy integrating detectors, but also to systems that they will have a photon counting detector. If we look again to the way dual energy is done in the clinic, you would have different solutions provided by different manufacturers. For example, Siemens on the left side here would have a dual source, dual detector measure system where you have two gantries, two imaging chains, and in that way they rotate around the body. They are 90 degrees apart, and they provide these two measurements. GE, for example, adopted a different solutions where you would have a single X-ray source and a single detector, but what they do, they switch the energy of the X-ray source, we call that KVP, from one projection to another. So you have this KVP switching technology. Philips on the other side, what I've done, they have created this sandwich detectors. Again, these are not photon counting, they're energy integrating, but they are in two layers in such a way that the first layer keeps the low energy measurement and the other layer does the high energy measurements. So once the data, the two measurements are provided one way or another by different manufacturers, what happens is that you take these images, and if you look here at these two clinical CT images, one would be the lower energy, the other one the higher energy. What you notice is that they are different. There is some contrast in the kidneys there, and it appears brighter, for example, in the low energy image. But in essence, those images are fed into some decomposition algorithm, a spectral decomposition algorithm to provide, in case there was contrast, for example, iodine maps, which would be distributions of the iodine in the body with the contrast agents that we typically use, overlaid on the virtual non-contrast image, sort of non-enhanced image CT. Or you can create this continuum of images of different, we call KEVs, or different energies that you can synthesize. And those virtually monochromatic images, for example, at low energies, they could have very good contrast. At high energies, they can solve some of the artifacts. So they have advantage to have them, all of them like that. On the other side, if we talk about photon counting CT, this is something that is new. All manufacturers are working on photon counting CT. All of them, they have some prototypes. For example, Siemens, they have modified some of their dual source systems to replace one of the detectors with a photon counting detector. So they have this hybrid geometry where one of the imaging chain, let's say A here has a black and white or energy integrating detector, while the other has a photon counting detector. And in this schematic here, they measure, for example, you can have 4 measurements, 4 energy selective images, and then they can go, for example, in material decomposition, and you can separate to create material maps for, let's say, iodine, water, calcium, and overlay them with different colors. Now, this is not only at the level of cartoons. Obviously, they have done, for example, studies in dogs. This is an example of using iodine and gadolinium injected in the body. Gadolinium was injected before, and it's cleared by the kidneys. And you see here in these images that you can separate the gadolinium from iodine. As well, there is a vial of bismuth there, another material that has a cage and could serve as a contrast agent. And so based on those 4 energies, you can create these color images. We call photon counting CT also color CT. As I said, this is not yet in the clinic. It's at the level of development. And for a number of years, what we have done is also to build our own preclinical prototypes. We started many years ago by building a dual energy CT system, a dual source geometry with 2 X-ray energy integrating detectors, obviously for mouse imaging here. More recently, we have built up to 4 prototypes. I'm going to show you the last 2 that they use photon counting detector. The first one is a system more or less like the hybrid system that Siemens has built, where we have replaced one of the detectors with a photon counting detector, while the other one is an energy integrating detector. The X-ray sources here, they are clinical X-ray sources. We have also another X-ray detector, a photon counting, which has higher resolution, and that one we have built another system for ex vivo imaging, spectral imaging. That being said, I'm gonna start talking about some of the challenges that we faced when we started building and adopting this new technology. let me go back about 3 years, and we had a collaborator, an industrial collaborator, the X-ray, and they were providing this prototypes for us. It's hard to see here, but there is a tiny stamp-like almost detector that has only 16 by 16 pixels, about 8 millimeter by 8 millimeter, a pixel size about 0.5 millimeter, which is pretty bad for small animal imaging, if you ask me. And so the challenge was also not only that was small pixel, that was not good enough pixel size, but also that the whole detector was very small. So we had to adopt this translate rotate geometry to be able to acquire projection images. And so you would translate the detector like that, which was positioned on some translation stage, and then you would rotate and get another angle. The scanning took quite a while. The detector had 4 energy thresholds, and I'm going to show you some of the images that we got there. Unfortunately, the detector was far from being perfect. You see here some what we call air images, so without an object. It's just imaging air. And you can see some of the pixels there in all the 4 energy beams, let's say. Some of the pixels are totally dark. Those are bad pixels. And some of them, they have a lot, not only that, but they are having a kind of a bad response, a biased response. So when we do that translate rotate scanning, what we were doing, we were translating this stamp size almost detector and trying to do a lot of image processing to kind of interpolate around those bad pixels. In the end, we end up with some images, some projections like that, which they don't look too bad. We have eliminated the totally dark pixels or dead pixels. However, there are still some bad pixels there. The pixels that they are not dead, but they have a biased response. If you would use these projections now to create sinograms, essentially stack them along the angle direction, if you look carefully here, and I hope the transmission is good here, you would see some vertical stripes in those sinograms. In a circular cone beam geometry, if you would do a reconstruction by filtered back projection, those vertical stripes associated with those biased detectors will create this ring artifacts, a very classical type of ring artifact associated with the third generation CT scanners. So there are ways to deal with these ring artifacts, but we have decided to look into deep learning for that. So this is our first case study, if you like, or application for deep learning. So how do we do it? Well, we adopted a UNet structure. We have the artifacted sinograms as input. And obviously for a supervised training, we will have to have labels or clean sinograms. I'm going to show you how we manage that. But in essence, we also scramble this artifacted sinograms to break correlations. And then we have low pass filtered, and the low frequencies, we pass them ahead. So we have a residual training strategy here. The UNet is just a classical UNet with 4 levels of resolution here, trained in this residual way. And then after that, the corrected sinogram is unscrambled, and we get hopefully the clean sinogram. Obviously, the first question is, we have to have some labels for this to be able to solve the problem. And for that, we use a strategy which is a combination of real data and simulations for training. Let me give you a secret. There is a way that you can avoid these ring artifacts by changing your sampling. Just not use a circular cone beam geometry, but do a helical geometry when you sample. The helical projections will still have those stripes, those problems, but when you do the reconstruction, you kind of smooth out those biased pixels, and the images, they look much cleaner. So that's one way with helical scanning, that you can provide some reconstructions that they don't have those ring artifacts. And then to create projections for the circular cone beam geometry, we then reprojected those in that geometry, and to create, in that way, we have created this labels that they are now not affected by the stripes that will give the ring artifacts. That was not all. We have used also energy integrating data that we have somehow added two simulated stripes, or we have sampled the distribution of kind of like the bias that we see in those bad pixels, and then simulated ring artifacts and created dirty sinograms through simulations. And a combination of this real data and simulated data have been used for training of that UNet. And actually, if we look at the results, it worked pretty well. We are very pleased with this. On the left side here, you see the 4 energy images, for example, without the corrections, and you clearly see the rings. If we pass our projection data through the CNN, through the UNet, you can see that we have corrected for all energy bins here. And that's also confirmed by the residual, the subtraction of the CNN, the no correction images. You can clearly see the ring artifacts here. But look at this also in a quantitative way. The structural similarity index matrix here is much higher for the CNN data. And then if you look at our experiment here, where we have imaged some mice, they have a sarcoma tumor here on the leg. These mice, they have been injected with gadolinium-based contrast agent, and you can see an accumulation of that contrast agent in the tumor. This is enhanced permeability and retention, but what is important is that the images on the right, they were corrected for the ring artifacts that don't show those rings, and also our way to estimate the accumulation of those gadolinium in the tumor is much more accurate that way. Okay, so that was our first application of deep learning. I would say in the image corrections. Remember that our first detector had pretty bad resolution. It was a 0.5 millimeter pixel size. That's quite bad for small animal imaging. But also remember that we have a geometry which is a hybrid geometry. We have another imaging chain that has a black and white or energy integrating detector that is actually a very high resolution by comparison. So why not exploit this? How about trying to combine the two sets to be able to get the best out of the two? So we get color from the low resolution photon counting detector, and we get resolution from the black and white detector. If you look, there is this concept of in image processing, which is called punch sharpening. They have applied it, for example, with satellite imaging, where they would have a camera that would be black and white but high resolution, and some camera which would be color but low resolution. And then the task there is how you combine to have both. In these simulations that we show here, let's say we are interested in looking at atherosclerotic plaque in the aorta. There you would have these 4 images which are low resolution, but they would have spectral contrast, and you would have the high resolution black and white image. So the task now is how do you combine the two with a similar to punch sharpening type of algorithm to be able to have both the high resolution and spectral information, so that eventually if you use contrast agents based on gold, iodine, that some of them they might be targeted, you would be able to look at that plaque calcifications or the uptake of different probes there. So for this, we have used a different type of network. This is a multi-scale, multi-depth kind of network. If you look on the left side, our input is a pair of a high resolution black and white image and a low resolution spectral image. This actually, they were sets that they were acquired with the energy integrating data, but switching or changing the KVP so that we get different types of contrast. And we have applied some blurring that would correspond to the photon counting data so that we have the low resolution spectral data matching somewhat the photon counting data. So if you look at the structure of the network, you have a deep network side where you can recognize maybe those inception module there. Essentially, you would have different scales, filters here from 3x3 to 5 to 7x7 and so on. You concatenate the features. That's also a residual way of doing this. So we have 2 inception modules like that, one after the other. And we also have a shallow network here. We took the idea from the super resolution networks, it turns out that this is better than just a skip connection here because it allows better performance, but you have 3 convolutions there on the shallow network. So the output here would be the high resolution image, but with the contrast of the spectral image that we had as an input. If you look at this phantom here, on the left side, you see that we have used 2 vials with different contrast materials. One would be gold and the other one would be iodine. The cradle here has a structure that shows pretty blurred because this is the way we can do it with the photon counting detector that we had at that time. But then after the palm sharpening, after using the high resolution black and white image, you can see much better detail on those structures on the cradle, and the information, the separation of different vials is still there. And actually, if you look at the distribution of those contrast agents, it turns out to be more accurate as well, closer to the value, the mean value concentration that those vials they had there. Obviously, it's not enough just to look at the structure or the details there, but you want to measure the modulation transfer function. So for the photon counting detector, you can see the red line here pretty bad in terms of 10% of the MTF here, about 1.4. But with the hybrid approach, the palm sharpening, you can see that the blue line here, it's at the level of the black and white image. So we are able to bring the resolution close to what the black and white imaging chain was providing. So this was another example of how you can apply deep learning, in this case, to kind of increase the resolution. You can maybe call it super resolution palm sharpening that we have done to improve our imaging. Another type of problems that we faced is that we would like to do dynamic imaging. We would like to do, and we have done plenty of cardiac imaging, for example, in the mouse. On the left side, you see a beating heart both on the coronal and temporal axis. Again, Darren Clark is the one that was driving this research. The problem with those images is that they are quite noisy, and this is just of the one of the energy thresholds, but as you will see, they can be even worse in terms of noise. What we typically do is that we, instead of using the analytical reconstruction, we use an iterative reconstruction that involves regularization. It's a sophisticated algorithm, and our regularization there is actually making some assumptions about the solution. One assumption is that the images, the CT images, are piecewise constant, and for that, for example, if we use bilateral total variation, for example, we can help solving the problem. The other assumption that we make in the regularization model is based on rank. So we want to have a low rank for those images, and for that we use singular value thresholding on patches. So in essence, we have this sophisticated image iterative reconstruction algorithm that runs on multi-GPUs and is using an optimization which is based on the Split-Bregman method that does a balancing between the data fidelity and the other 2 regularization term to achieve the images on the right side, which they keep the motion and they are also much less noisy. The problem with this is that for reconstructing this 10 cardiac phases set, typically it takes at least 2 hours. So it's a long time. In this situation, we looked about maybe we can find a data-driven regularization model through deep learning. Maybe that can be used to replace our regularization models in the iterative reconstruction, or who knows, that regularization or denoising may be applied to the analytical reconstruction to clean it up somehow and replace completely the long lasting iterative reconstruction, which, like I said, it takes about two hours. So obviously for that, we had to have some, again, we go for a supervised training approach. We have to have data. We have used a different detector. This is a Santis by Dectris. We have acquired plenty of projections, about 9,000 projections with helical scanning. and with very short exposures because we want to image the beating heart. The heart in a mouse beats about 10 times faster than humans, so 10 milliseconds exposures are what we need for that. The dose is very low as well by comparison with our prior work. So 190 milligrade for micro CT at this resolution is quite low. Now, if you look on the left side, we have this fully sampled beating heart images, which they look noisy. They are reconstructed through analytical reconstruction. If you plug them through the iterative reconstruction with regularization, you get them clean like we've seen before. And those will be our labels, the ones in the middle. On the other side, however, we would like to create input images which are even noisier than our fully sampled reconstruction, because we aim towards finding a solution that is robust and also reducing the dose even more. So what we have done to create those input images, we have undersampled our set of projections to have equivalent of quarter dose scans. So that's why they look so noisy, because essentially they have been reconstructed with 1/4 of the number of projections that we have used for the images on the left side. The structure here for the unit that we have used is, first of all, it's adapted for 4-dimensional imaging. So we have 3D patches plus time. In this case, time is the 10 phases in the cardiac cycle. It's a UNet, but also because we have constraints related to the amount of memory that you have on the GPU. On the Titan XP, you have 12 gigabytes. So we had to create these asymmetric unit structures that will be able to fit the data during training. And also we have spatial max pooling and temporal max pooling here and unpooling. So there is both time and temporal pooling operation that they happen. And also something interesting is that we have a residual connection here, but what we do use a DCT, discrete cosine transform, and we send through the skip connection, we send the like the low frequencies to the end there. So this worked well. I'm going to show you some examples soon, but again, if you look at some sets here, I'm giving an example of 5 mice that they were used for training. For example, you can see the fully sampled algebraic, but not regularized reconstruction. You can see the iterative reconstruction, our level labels on the middle row here, and you can see the undersampled algebraic reconstruction, which are the inputs to our network there. The results are shown here. You can appreciate how noisy the input images they were. Look at the last image on the right, and that corresponds to the last energy threshold from 55 keV to 80. And you have very few photons there, so you get very, very noisy projections. However, the network is able to do pretty well. It's not perfect, but it is amazing the amount of denoising that it's able to provide. Now, this is actually 5-dimensional imaging. So we talked about the temporal axis plus the spatial 3 dimensions. But remember, we have 4 energies, right? So energy plus time plus 3-dimensional space. So we can do decomposition of those, spectral decomposition. What I'm showing on the left side here, this is a mouse that is a model for atherosclerosis. It's an ApoE knockout mouse. What you notice is that there are some tiny calcifications that we are able to resolve, and also you can see the beating heart. So this is opening, we can do cardiac function estimation, but also looking at calcification or plaques, or maybe plaque composition, for example. Finally, I would like to switch now to a clinical application. This is for a clinical dual source system. Again, the major contributor here was Dr. Clark in my lab. What you see on the left side is the clinical system, the Siemens clinical system. Remember, it has 2 imaging chain. One of the detector, both detectors are black and white, but one of the detectors is larger in size. The other one, because of constraints that they relate to the mechanics of the gantry, it's smaller in size. Now, the problem that we're trying to solve here is the fact that to be able to do a spectral decomposition, you can only do it on what we show here, the blue circle, where you have information from both imaging chains. But in some cases, when you have the patient not well positioned or you have some large patients, it is possible that the 33 centimeters, which is the size of the small field of view, there is not enough to cover the anatomy. And you can see here that some of the lesions in the liver there, they are not covered and they are not decomposed. In red here would be the iodine mop. And there is a lesion there that is not decomposed. So our task here was, can we do a deep learning solution that is like solving a data completion problem, trying to extrapolate that spectral information to the size of the large field of view so that we can cover everything. For that, again, a supervised training strategy, we have to have data that we can use as labels. And we have adopted the strategy where in order to have the truth, instead of using the blue, which is the small field of view, we use some training masks which are smaller in size, like the red ones, and also are variable in size. So the network will try to estimate contrast between the red and the blue. There we have information, the truth from both imaging chains. And by learning to extrapolate like that, then it will be in the future when we'll use the whole blue as an input, we will be able to extrapolate also in between blue and gray here, which is the real case that we face in practice. The network here, it's essentially a UNet which has another layer at the beginning that is a transformation layer or piecewise linear transfer function kind of layer that we have built. We call the small imaging chain the large field of view imaging chain A and the small field of view imaging chain B. What we do is that we try to estimate first through this layer the contrast of B from A. That is not perfect, but is a good estimate for the beginning. And then we kind of pad that information to the true B, and we feed that to the network, the UNet here, together with the XA here. The UNet here is a 5 levels of resolution UNet. The output, we call them YA and YB. The goal is to really look at the extrapolation YB, while the YA would hopefully stay the same. Some results here, we have the two images, XA and XB. This would be the outputs. YA should be exactly like XA, and it looks very much the same. And the YB here in the center, we have the true B, while on outside of this red circle, we have the extrapolation that the network provided for the YB. Now, if we try to look at this, in a more quantitative way, if you would subtract, for example, XB and XA here, which would be the initial contrast between the two imaging chains, and look at the contrast in between the red and the blue, you can see that, as expected, the contrast is different. If we look at after the piecewise linear transformation layer there, you can see that that difference is reduced. Ideally, it should be 0, but it's only made small enough after the second UNet, let's say, which brings that almost to 0 to errors of about 4 Hounsfield units or 6 Hounsfield units, which is very, very little error there. Again, for some cases that, in this case, for example, the patient was placed on the table in a kind of off-center way, and you can see that we can estimate outside of that yellow circle, and again, a large patient where traditionally we will not be able to say anything about the iodine map for that lesion there at the bottom left where we have the arrow, but through the extrapolation, we're able to have that. I will end up now. I hope I have convinced you that there are ways to use this deep learning in this image formation, image denoising, extrapolation that we have used it for. Sometimes this is not, it's still fixed. We need fixed protocols or way that we have acquired the data have to be reproducible. And a lot of work for deep learning should go into finding ways to deal with that, making the solutions more robust. We have applied lots of deep learning to other problems like segmentations for our co-clinical trials. I'd like to end here. Many people that I I have to acknowledge, especially Darren Clark and Matt Holbrook, E.T., Ketan Gagada at Texas Children's that has provided for us contrast agents, and we are very grateful. Clinical collaborators like Daniela Marin and Fitz Schwartz, as well as David Kirsch and Yvonne Mori. I acknowledge my funding and also the Dectris, the manufacturer of the X-ray detector photon counting detector. Thank you.
Thank you very much, Christian, for the excellent talk. It's actually quite staggering to see some of the improvements you've been able to make, and hopefully we'll be able to see that used in the clinic in the not too distant future. For the sake of time, I want to invite the audience to enter questions into the Brella chat and invite Dr. Badea to jump over there, and he can answer your questions directly there. But for the sake of time, we'll go ahead and move on. Ahmed and Brett, I'll invite y'all to go ahead and turn your cameras and microphones on. And I'll turn it over to you, Ahmed, to make the next introduction.
Great. Thank you so much, Susan. Yeah, so we're excited to move on to the next speaker. We have Dr. Brett Scholson, who is a principal application engineer at MATLAB. He's going to be talking to us about, his talk is entitled From Pixels to Models, an Introduction to Image Analysis with MATLAB. So thank you so much, Brett, and we're excited to have you start. I guess you can share your screen. Oh, it looks like you're doing that. Okay, great.
Okay, I'm sharing. So can you guys hear me? Can everyone hear me?
Looks good. Yes. And sounds good.
Okay, great. So thank you everyone for joining me. As mentioned, my name is Brett Scholson. I'm a principal application engineer with MathWorks. I've worked for the company for 15 years, but I've been using MATLAB for image analysis for over 25 years, including for my dissertation in quantifying retinal blood flow. During the 15 years that I've talked about image processing and image analysis, the architecture and the landscape for image analysis has changed dramatically. It seems that we can't have a conversation these days without the conversation turning immediately to deep learning. Deep learning is transformational, and if it's the right tool for the job, there's probably nothing that's going to outperform it. But it's also true that not every image analysis problem is a deep learning problem. So in a whirlwind presentation, I'm going to give you kind of an overview of image analysis capabilities in MATLAB, starting with what we call pure image processing, leading into computer vision, then through traditional machine learning, and finally ending up with deep learning. And as I go through this, I'll talk a little bit about how I differentiate between those clusters. Okay, so MathWorks, we have a tool called MATLAB. MATLAB stands for Matrix Laboratory. An image is nothing more than a matrix of intensity values. And if you look at an image at the pixel level, that is element by element, and you start doing quantification at the element level, you're in the realm of what we often think of as pure image processing. I have an example here. If we have an image, unfortunately my example is not really medical flavored, but hopefully you can take the leap of faith with me. I have an image here called rice grains, and if we wanted to look at this and ask a question, for example, how many objects are there in this image? And I'm purposely saying objects, not grains of rice, and I'll make a point about that in just a moment. But we can do this with a traditional image processing bent. And typically, the first thing we do is we want to create what we call a segmentation mask. We want to binarize the image in this case, making the foreground, the regions of interest white and everything else black. And this is, again, classically an image processing problem. Here, I'm going to actually step through this. I'm going to just read in an image and visualize it. Okay, let me make my screen a little bit bigger here. And again, we ask a question, how many grains of rice are there? And the first thing we want to do, as we say, is to binarize this image or to segment it. There's lots of ways to do that. So I'm going to come up to my apps toolbar. We have a lot of tools that extend MathWorks tools, a lot of different toolboxes. On the image processing computer vision tab, I have several things that might be useful, but in particular, if I want to segment an image, I might come up to the image segmenter app, and I might just launch image segmenter and pass in my image. Let's just launch that real quick. Okay, and I bring my image in, and there's lots of ways, as I say, that we can segment this. Probably the easiest thing to do, most fundamental way of segmenting is to create a threshold. You say anything above a particular threshold value is white or true, anything below it is false. So I could just say, let's threshold it, and MATLAB will automatically take a stab at providing a global threshold. That doesn't work particularly well because of the difference in illumination across this image. So I might instead try a manual threshold, and I can tune it. You can see how these tools are interactive and make this process easy. And fundamentally, when I come down to adaptive threshold, we can adjust the threshold automatically relative to the local background intensity. And when I like what I see, I can create a mask, but more importantly, this tool and all of our tools also generate code. So if, for example, I generate a function, MATLAB writes for me the code that will allow me to automate that process. If I have many more images, I don't have to do that manual UI level interaction each time. And you'll actually see that in this case, I'm really just binarizing with an adaptive threshold, so you get the equivalent code. So I'm not going to save that. I'm just going to come along and say, let's binarize with an adaptive threshold. And now I get this segmented image. Next thing I want to do is to count the images, count the objects. And I'm saying let's count objects, not grains of rice, because if I were to look at this, you'll see that if I just counted blobs, I'm going to over segment, I'm going to over count, because I have all these little blobs in here. So how do I proceed? Well, if I went to my doc and I typed in, how do I remove small objects from an image? You'll find that there's lots of ways to do that as well. The easiest way is just to call a function called BW area open. I'm going to say anything less than 50 pixels, 50 contiguous pixels, let's get rid of. And when I run that, I now have a very clean mask of my image. And then counting the objects, again, I can go to my doc and say, how do I find objects in a binary image? I'm just going to count connected components. If you want to talk about this, we can have a conversation about what this is doing afterwards. But if I run this, I get a connected components object, and I can see that I have 93 objects, 93 blobs. And that's a pretty easy thing to do. It's a pretty fundamentally image processing operation. One more thing, though, if I pop this out and zoom in, you can actually see that there's another problem in that these contiguous grains of rice here are treated as a single blob. So I might then, for example, go into another tool called our Image Region Analyzer tool. I can pass this in, and I can do things like sort by area. And you can very easily see that you can find a threshold, a size threshold above which you have single grain, a double grain, and below which you have single grain. So I could then come along and, for example, filter and show me anything with an area greater than, let's just say 250. And now I have another image that I can just count. And if I add the count of this image to the count of the single grains, I get a better representation of the number of grains. Okay, so all that said, that is a pure image processing workflow. I'm treating the image as a matrix of intensity values, and I'm looking at them pixel by pixel. I haven't really done any kind of qualification of what these grains are or what these objects are. And to do so, we get into kind of the next step of thinking of an image not just as a collection of pixels, but as a collection of features, looking at the local relationships between pixels. And with those relationships, with this concept of features, we can ask higher level concepts, higher level constructs from our images. For example, if I have a 3x3 matrix, if I just simply thresholded that in an image realm, I could look at that and I could say, hey, there are 5 pixels, 5 super threshold pixels. That differs from how we look at this in a computer vision world. In a computer vision world, we would look at this and say, for example, we interpret that as the letter L. Okay, so this idea of interpretation, of using features, brings us from pure image processing into the realm of computer vision. With computer vision, we use this idea of feature extraction, and I like to think of it as a sparsification of our images. Here, for example, if I had a high density image or representation of a bicycle, I could do image processing analysis at the pixel level, or I could look at, for example, a histogram of oriented gradients, hog features, and you can see how with this very simple transformation, I can operate on the image as a collection of hog features rather than a collection of pixels, or even surf features, speeded up robust features, where in the computer sees mathematical representations in our image that we don't necessarily see, but that are representative of the type of object that we're looking here, in this case, a bicycle. And if we cluster a bunch of bicycles or a bunch of bicycle parts, we can create what we call a bag of words or a bag of features. And we can move kind of from the realm of pure computer vision to the realm of classical machine learning. So when we use this, when we go from image to feature space, we typically use what we call a detect, extract, match workflow. That is, we have one or two images, we detect features in the one or two images, we extract properties and we match them. And from those matches, we can do some pretty amazing things that are very, very difficult to do in image processing, very easy to do in computer processing. So let me come along to back to my MATLAB and run a quick example. If, for example, let me just clean up here. If, for example, I wanted to detect an object in an image, let's just say I have an image of a staple remover, and I have a cluttered scene. And my goal here is to say, how can I use this target to find a staple remover in this? And again, I recognize these are not medical images, but these could be tumors, these could be any kind of medical object that has a characteristic feature space to it. But though this is very difficult to do in an image processing realm, I can run this kind of detect, extract, match workflow. So I'm going to detect features in both of those images. Here I'm just going to visualize what those features look like, and you can see that I've got features calculated in my staple remover image. I've got features calculated in my scene image. Some of those features are the same. And what I'd like to do is extract properties and match those features. So a few lines of code, I get it. I show my matches. And you can see the next problem in that some of those features are mapping directly to the staple remover in this box, despite the fact that these are at different scales, different orientations, different rotations, everything. But I also have these outliers. And if I were to create a computer model to map from this space to this space, I would have probably not a very good model. What I'd like to do is I'd like to estimate a geometric transformation that casts from this space to this space in a way that is robust to the idea of outliers. And the way that we do that is with a workflow called RANSAC, Random Sampling and Consensus, where I subsample the matches, and I iteratively subsample until I find a low noise model. The nice thing is, if I say I just want to estimate a geometric transformation, that RANSAC operation is done for me. And if I now show the matched features again, you can see that those outliers are gone. All I need to do now to find the staple remover in the scene is to find the location of the original box in the target image and cast it forward with the transform for points forward command. And you can see I've found that object in the scene. And that is a classical computer vision approach. We use the idea of features. Features are much easier to use, much faster to use because it sparsifies the image. And I'll just show you here that really all of that, everything I showed you, there's a lot of visualization in there, but everything I show you here is really summed up in about 10 lines of code. So we've kind of moved from image processing to computer vision. And the next step is how do we go from operating on two images to operating on multiple images? And the first step is via a classical machine learning approach. Now in this graphic, I've got a few images that feed into a manual, what I call a manual feature extraction bucket here. What I mean by that is, in the previous example, we used surf features. I'm going to explicitly tell the computer to use hog features or surf features, or whatever kind of features I want to tell it. And then based on those features, I'm going to say explicitly or implicitly, whatever, I'm just going to constrain the problem, and I'm going to tell the computer to create a support vector machine or a decision tree or some kind of computer model. And based on those specific, the constraints, with a little bit of images, a little bit of data, ground truth data, I can create a model that often works very, very well. So if, for example, I had 5 objects that I wanted to differentiate, again, not medical, but these could be 5 different medical classes. If I had enough ground truth images, I could differentiate them with a classical machine learning approach using the idea of a bag of features. Let's calculate, for example, surf features across all of our data, and then create a dictionary of what each feature set looks like for each of these different models. Now, I don't have time to run this, but I would show you what it looks like in code. I would just very simply wrangle my data with an image data store command. I can count them. And here I'm just going to show you a table that says, with just 15 images in each of these buckets, I can create a machine learning model. Now, 15, it's not 15,000, it's not 15 million. This is not at all deep learning. If I had 15,000 images of these, I would probably go to a deep learning model. But with a very few images, which means a very easy on-ramp as far as ground truth, as far as computational expense, I can very easily split the data into a training set and a validation set, create my bag of features, train an image category classifier, and then I just simply read in an image from my validation set and predict its class membership. So it's a very small bit of code with which I get these kind of telltale or representative dictionaries of features for the 5 different classes. I read in an image, I calculate its visual word occurrence, its feature space, and mapping back to these 5, I say which one of those feature spaces is it most like, and I can then characterize that object, in this case as a MathWorks cube. I can do that across the board. It works reasonably well for a small set of images. What we'd like to do next, though, is to say, what if we have a lot of data? What if we have thousands of images or tens of thousands of images? What if we want to drive down the road with our hands off the wheel? What if we want to look at medical images and make very, very good medical diagnostic capabilities? I can't do that with 15 images generally. If I ground truth a lot of data, we're going to differentiate machine learning from deep learning by saying, I can create a model that's going to predict for me what my feature space is in a way that's very, very unconstrained. So in the machine learning space, we said we're going to create surf features and we're going to create a support vector machine. In the machine learning, in the deep learning realm, we only specify the architecture of the model. And we do what we call end-to-end learning with a lot of data, ground truth data that's potentially very onerous to get. We can help you with that as well. We're going to tell the computer to figure out what it needs to know from those images to calculate our output space or to classify our data or to do our regressions or whatever it is that we want to do. And because we're not constraining the model, where machine learning typically will saturate, there might be only so much that we can get from surf features or hog features. The deep learning algorithms scale with data, that is, they do not saturate. So the more data we give it, the more ground truth data we give it, the better the model gets. So ultimately, we hear about deep learning these days. Deep means there are a bunch of layers. We can stack the layers linearly or non-linearly. I think of the layers as kind of functional layers that we can pass information through. When we talk about deep learning for images, we often think about neural networks, convolutional neural networks, in particular CNNs. I'm not going to have time to go through what all the layers are, but you'll see a bunch of repeated units, often convolutions, ReLUs, and poolings and normalizations. You'll see these often over and over again. And then when we get to the end, we have a terminal stage that captures everything above it, creates a kind of a probabilistic layer, a softmax layer, and then uses that to map into the classification space. Now, I'm not going to talk about all of these. I just want to talk about one. Why do we look at convolutions? Well, convolutions are very, very good at looking for patterns in images. If, for example, I wanted to create a model to differentiate the numbers 0 through 9. These are digital, obviously not handwritten, but if I were looking at the patterns that represent this number 0, the patterns here, these are the convolutional kernels that would match or map to this letter or this number. And those patterns are typical of what a 0 would look like, and they're very different from what a 1 would look like. Now, in classical machine learning, we might say these are the kernels that we want to look for, In deep learning, we say, create some convolutions and iterate until you create the kernels that make this the optimal. It's an optimization problem that optimally differentiate between a 1, a 0, or all their digits. Okay, all that said, there are two typical approaches to doing deep learning. One is kind of the gold standard. If you have enough data, enough ground truth data, you go through, maybe you label 100,000 images, and with a high-powered GPU, you let your computer churn away, and it churns for days and nights, maybe, however long it takes. The training aspect is very computationally expensive. It could take a long time. The inference stage, the using the model that you've trained already to make a prediction, can be blisteringly fast. It can be fast enough that we can drive down the road and make our predictions.
Oh, just wanted to give a quick, I guess, update that, you know, we do only have a few minutes left for the end of this talk. So
almost done. Thank you. Okay, and then this is the gold standard, started from scratch with a lot of data. This is not generally how we often do our deep learning, though. Instead, we use an example like a deep learning model that's pre-trained, and we do what we call transfer learning, just modifying the architecture a little bit and retraining it using the features that it's learned on millions of images to do a different classification problem. And what we get is a very, very unconstrained, amazingly good model that can look at objects that the computer has never seen before and map the visual space of mouse or laptop to representations of other mouse, laptop, sunglasses that it's seen before. Works really nicely. And in conclusion, I'm going to just say, I want to close with a slide from Gartner. Gartner is an independent research and advisory company that recently recognized MathWorks as a leader in the field of computer vision, deep learning and machine learning. So completeness of vision on the bottom versus ability to execute. We're up here in the leader category. We'd very much like to help you with your deep learning problem. I'm going to share all this information with you. I just wanted to share Angelo's name. Angelo D'Amato is one of my colleagues at MathWorks. Here's his contact information. If you want to have deeper conversations about any of this, please contact Angelo, and he'll get you in touch with me or with he'll marshal the resources to bring this to bear. So thank you very much. Appreciate your attention.
Great. Yeah, thank you. And yeah, since we're running a little bit late on time, maybe we'll just take one question and then move on to the next talk. Is there a question for Brett?
So there currently aren't any entered, but we'll invite the audience to go ahead and enter questions in the chat umbrella. And yeah, Brett, if you can just jump over there and answer any of those that may show up, then we can go ahead and move on to the next speaker.
Will do. Thank you very much.
Great.
Thank you. Great talk.
Thank you so much, Brett. Really appreciate it. All right. Well, it's my pleasure to introduce our next speaker, Professor David Fuentes, who is joining us from MD Anderson Cancer Research Center. He's going to be presenting a talk on entitled Imaging Informed Machine Learning Models for Low Grading of Gliomas.
Yes, hello. So are you seeing my
Yes,
my PowerPoint view and not my presenter view?
We see the slides. Thank you.
Okay, and my audio is okay?
Yep, sounds great.
Great. Okay, so I'll begin. So thanks for the introduction. My name is David Fuentes. I'll be sharing imaging-informed machine learning models for local grading of glioma today. So first and foremost, I'd like to thank my interdisciplinary collaborators. This work has really been enabled by a multidisciplinary collaboration across neurosurgery, neuropathology, neuroradiology, as well as imaging physics. In particular, the graduate student, Evan Gates, has been really doing a lot of the heavy lifting for the analysis that I'm going to show you today. So a bit of an overview background. Our motivations for this talk are really to improve the outcomes for glioma patients. The key takeaway message from this talk today is that I want to convey is that we really have a very unique data set for training our machine learning models. We have very tightly correlated imaging with pathology within 1 millimeter accuracy. We know the exact correspondence between voxels within an MR image and pathology data within a glioma to train our machine learning models. The key results, we believe we're accurately able to use this machine learning model to locate aggressive disease. And then our current work that I'll show you today is we're using this to extend to large scale neuroimage analysis studies to further validate our work in machine learning. So I'm the guy that brings a random forest model to a neural network party. For historical reasons, we really started working on this project around 2013, 2014, when tree-based methods, random forest methods, were still winning a lot of the crowdsourcing challenges. So half of the machine learning models within our workflow are still very much random forest based because they're getting good accuracy, and we really saw no need to move from a working code to a neural network just because it's trendy or whatnot. However, I would like to echo some of the points from the previous talk. We have been using more and more neural networks within our neuroimage analysis workflow, particularly for image segmentation, segmenting within the brain, the skull stripping, the brain tumor, resection cavity, ventricles, and brain structures, and so forth. And we found the MATLAB workflow to be particularly helpful. So in particular, the analyze network function, a common theme that's come up is if you're getting started, how do you start incorporating neural networks into your workflow? So I found this analyze network functionality within MATLAB to be particularly useful. It goes through and describes in detail every layer within the network. And my students and I typically start from a ground up approach, and we look at the fundamental mathematical structure of each layer and build that up from the vector spaces input to the vector spaces output. We really haven't seen any, so we started off doing everything with TensorFlow, but we really haven't seen any difference in performance-wise between MATLAB and your favorite open source package. Everybody calls CUDA underneath the hood, so it's been very helpful. And then one more further point is just the commercial versus open source. The ability to contact the MATLAB help desk when you get really stuck on a problem has been really helpful for us. And then as far as onboarding new people in the lab, the deep learning on-ramps and the machine learning on-ramps have been particularly helpful for conveying the fundamental concepts and translating the concepts into programming. So our overall motivation for this machine learning project is to improve outcomes for glioma patients. So the grade within the glioma is a particularly important quantity. Grade 2 patients have an expected survival of around 12 years, whereas grade 4 patients, higher grade patients, glioblastoma patients, have an expected survival of much less, 14 months. And so importantly, within the clinic, typically a tissue sample, a small set of pixels within this MR image are sampled, and that tissue sample is sent to pathology, and a pathologist looks at it under a microscope. And from there, the pathologist assigns a grade to the tumor based on the highest grade found within the tumor. This is an invasive approach. The skull has to be opened, the needle has to be inserted into, or the tumor has to be resected, and tissue has to be excised to assign a grade to a tissue. Our fundamental hypothesis from our machine learning work is that we can use a very unique data set of imaging pixel-wise correlations with pathology, tissue pathology, to develop a surrogate model to help guide where the high-grade disease is. So as was pointed out in a talk earlier this day by Dr. Jia Wu, the tumor is known to be particularly heterogeneous, right? So if I have and then the patient may have multiple grades within the tumor, right? So here For example, a high-grade disease may be present here, grade 4, but if you biopsy here, the pathologist may return a grade 2 disease to you. And so if the highest grade disease found in the tissue samples was grade 2, a patient would be so-called undergraded and would be treated less aggressively accordingly and ultimately do some harm to the patient. So what we've been working towards doing is developing a pixel-wise correlation between imaging and excised tissue pathology to provide a heterogeneous map, a spatially varying map of grade throughout the entire tumor based on a random forest model. So the unique data set is basically a stereotactic biopsy that is used to sample a piece of brain tissue within the accuracy of the stereotactic guidance systems within the neuronavigational system within our brain lab suites at MD Anderson under neurosurgery. And importantly, we're able to register the location of the known biopsy location where we have tissue pathology information and register that with the 3 or 4 pixel neighborhood that corresponds to the tissue pathology. So we can have a complete image characterization of the tumor and correlate that with the tissue pathology to build our machine learning models. So ultimately our data matrix looks something like this. Along the rows, we have a row that represents a particular tissue biopsy sample. For a particular tissue biopsy, we have a known grade. Grade 2 would imply that the patient would have a longer survival than grade 4 GBMs and so forth. And then along the rows are input-output pairs, our input imaging, output predicting pathology. The imaging data is fairly comprehensive. We have the full suite of the advanced brain tumor imaging protocol at MD Anderson. The anatomical includes your usual suspects, T1, T2, FLAIR, et cetera, diffusion-weighted imaging, perfusion, permeability, DCE, with a grand total of 24 total distinct images arriving from the derived maps from this more functional data. And then we also have a fairly comprehensive list of pathology information at our biopsy sites, where for this talk, we're focusing on just the grade of the tumor, how aggressive the disease is in some sense, grade 2, 3, or 4. And at each point where we have a biopsy site for training our random forest model, I've labeled here in green and red. These is a brain template where each patient within the clinical trial who had a tissue biopsy was registered to this template. And here, green means this was an actual tissue tumor biopsy site, and the corresponding contralateral red side is a normal tissue, so-called, quote, virtual biopsy, which we can't, for ethical reasons, can't sample normal tissue. This is just a contralateral to balance tumor with normal tissue, the contralateral side of the imaging side. So we have balanced tumor, not tumor, for training our machine learning models. So to get to the punchline, we're seeing good accuracy with our machine learning random forest model voxel-wise. So voxel-wise prediction of input imaging, output pathology, we're seeing about 95% accuracy. This analysis follows the conventional radiomics type pipeline that we saw again in Dr. Jia Wu's talk. We do a univariate analysis to assess, make sure that we have inputs that correlate with outputs so that we have some hope of building a predictive model, do some variable reduction. And then ultimately the inputs to our model are a T2 image, an ADC from the diffusion, a K-trans from the DCE, and a CBB from the perfusion image. We did a 5-fold cross-validation accuracy to validate, to provide some accuracy estimates of our machine learning model performance. And then here's just a kind of a confusion matrix of what we're seeing. To further that point, if we look at just our machine learning model, or random forest model for separating higher grade disease from normals and lower grade disease, we just want to get an idea how accurate we can identify this high grade disease. We're seeing about, for ROC analysis, we're seeing AUC of 0.98, and for precision recall curve, we're seeing AUC of 0.90. So, I mean, the moral of the story is that we basically are at a point where we're confident that we're getting accurate pixel-wise measurements. And so if we have pixel-wise measurements, then we can raster scan across each pixel within a 3-dimensional volume and generate spatial maps of high-grade disease, normal tissue, and lower-grade disease, grade 2s versus grade 4s, right? And so this is a pictorial representation of the model that we built. We have input T2, ADC, CBV, K-trans into our model, and then the output prediction is this green, red, and yellow image that effectively provides a bullseye, a target for clinicians, right? So if a neurosurgeon would biopsy the red, this is the higher grade disease, or resect the red, this is higher grade disease, and then radiation oncology would deliver radiation to this high grade disease as is the goal. So to further this analysis, we're at a point where we get a map of the high-grade disease within a brain volume. And so we wanted to further validate this analysis within a large-scale study, if you will. So our colleagues in the neurosurgery department, in particular, Dima Suki, has maintained a phenomenal data science resource for neurosurgery with a long history of all the neurosurgical cases, and was able to find for us 2,500 distinct glioma cases that we could use to validate our models. Within this 2,500, only 1,200 of them, approximately 1,203, had the appropriate imaging input that we needed to use to build, to apply our models to attempt to validate. So here the idea is that for a grade 2 disease, if we apply our model, if we apply this spatial map to each of the 1,200 patients, we'd expect to get some volume estimates of lower grade disease versus higher grade, more aggressive disease. And because this data has survival data, we could stratify lower grade disease based on the expected survival of grade 2s versus grade 4s, and expect to see some separation between aggressive disease and less aggressive disease if our models are appropriately working correctly. So to scale from the fairly limited number of patients for our random forest model to a large-scale neural image analysis study, we really had to implement our data analysis pipeline on the high-performance computing clusters at MD Anderson. An overview of our pipeline is that we basically build a relational database for the neurosurgery database that our colleagues provide us. We use queries to identify the SQL type queries to identify the T1, T2 images, et cetera, that match our needed input, convert DICOM to NIFTY, bias correct, skull strip, register everything to a common template. Neural network segmentation algorithms are applied to segment the tumor, the cavity, the resection cavity if it's possible, normal tissue structures, and ultimately we do some intensity standardization so that we normalize the MR image to make it quantitative for our analysis, and then ultimately apply our grade prediction pixel-wise throughout the brain to assess the high aggressive disease burden. So many labs have done large-scale image analysis. What really distinguishes this work is that we have a data QA tool that was developed by Evan using the R-Shiny package in particular. And you can think of this as kind of a Google Maps view of our data. So if we have a publication ready figure here, we get some outlier. We can quickly zoom in from the publication figure and get some intuition on what's going on with the data. So it has all the anatomical images displayed, and it gives us some confidence that the images are registered, that the neural networks are giving us accurate skull stripping, accurate tumor mass, and accurate ventricle segmentation, all as input, all as intermediate steps within our data processing pipeline, so that we can build some confidence that if we're making a prediction, that we can put money on the table that we actually believe in that prediction. The intensity values are provided here so that we can get some kind of quality assurance that our intensity normalization for making our images quantitative is meaningful. And then here we just have a kind of a dashboard showing predicted tumor volume on all 1,200 patients versus the manually measured tumor volume from neurosurgery to see if we find any outliers with our neural network segmentation algorithms. So ultimately, we found that the quality assurance to be very important. In particular, there were some cases where, or one case in particular stands out, where a spine image showed up within the neuroimage analysis pipeline. It passed all of our queries, didn't segfault on any of the bias correction, the neural network, and got a result, and showed up within a survival analysis plot. And if we didn't have these quality assurance tools, it would have slipped through. So very important to quality assure your data, and just important to look at every one of them to make sure a sanity check that you have high quality data in your final results is just kind of good science. So our final result is that when we make our predictions using this high performance computing pipeline of high grade aggressive disease versus lower grade disease on this retrospective cohort of 1,200 patients, we're actually seeing some separation, that the higher grade disease predictions have lower survival than the lower grade disease within this database. And we're seeing a hazard ratio of about 1.67, and for reference for literature values is about 1.7 for extended resection and tumor. So in summary, we believe we can accurately localize the tumor grade and estimate it non-invasively, not to replace biopsy or anything, but just to kind of provide a surrogate for guiding biopsy and resection as a machine learning tool for guiding, for image guided therapy. And we've retrospectively validated this in a fairly large cohort, and we're at a point where future directions are prospective validations of our pathology predictions. I'd like to thank the NLM Training Fellowship in particular that has provided Evan support for this data analysis and data curation, and support for his time. And with that, I'll finish and take any questions.
Yeah, thank you so much, David. Fantastic talk. You know, just as a reminder, if people have questions, please enter them into the chat. And David, if you wouldn't mind addressing that in the chat, that would be fantastic. And, you know, we're still running a little bit behind, so I'm going to introduce our next speaker. The nice thing is there's a break that would allow us to make up some of the time. So hopefully we can get back on schedule at 3:05. But our last speaker for this session is Professor Mary Ellen Gigar. She's from the University of Chicago, and her talk is entitled Machine Intelligence in Medical Imaging, Breast Cancer, and COVID-19. We're very much looking forward to your talk, Mary Ellen. I see you have your slides up. So.
All right. Can you hear me okay?
Yes, thank you.
All right. Well, thank you for the invitation. I'm happy to be here. A great program. Let me just move along. So here are my funding and conflicts of interest. A lot of grants in cancer as well in COVID-19 now. I have some conflicts here. One involving the translation of our research to clinical care. And it's the policy of University of Chicago that I present you this. So my talk is on AI and medical imaging with examples in breast cancer and COVID-19. And the focus here is for us to use images that are clinically and routinely obtained on the population. We want to ask questions about the relationship between features seen in the medical images and the biology of disease, so that eventually we can detect and diagnose disease early and give the right patient the right treatment at the right time. We also want to improve the efficiency and the workflow of medical imaging interpretation. But what is medical interpretation? Medical images are meaningless grayscale, color scale patterns unless viewed and analyzed by an intelligent observer. That might be the radiologist, the computer, or a combination of both, as in CAD or AI aided. And the task of the human eye-brain system is to find and locate a signal in an image, characterize or classify or diagnose that signal relative to disease or not, or clinical decision-making on patient management through integrated diagnostics monitoring. And the tasks of AI are basically the same. But here we convert images to quantitative values, and we proceed, noting that's very important to know the clinical task. I like to take this moment here. Often I hear people talk about CAD and AI in the same sentence in the wrong way. And it's really important to know that AI is the computer algorithm, whether it's radiomics, human engineered, machine learning, deep learning, that exists in a system for various types of implementation in clinical practice. And CAD is one of those examples. But CAD is a computer-aided detection or computer-aided diagnosis, can be used as a secondary reader, or even more recently as a concurrent read. But AI can also be used in a primary reader setup. Sometimes it's being looked at as a triage situation or a rule out, where it identifies some cases that perhaps won't need to be looked at by a human. And then there's completely autonomous AI systems, but I think it will be a while before those, because somewhere along the line, there's an end user. And this setup here also does not specify whether the AI is to help interpret an image or to also integrate it with other diagnostics. AI can be applied for many medical imaging decision-making tasks: risk assessment, screening, diagnosis, prognosis, treatment planning, assessment of response, patient management, as well as disease discovery. And there's many challenges of AI in medical decision-making. You need to understand what is the clinical task you're addressing. And is AI being looked at as an additive component in the decision-making or as a replacement? Or is AI being used to improve performance, say of radiologists, or to improve interpretation times for efficiency? And it's also important, you develop the AI algorithm, but besides looking at the performance of the algorithm, you need to look at the performance of the end user who's going to be using the output of the algorithm. Is the AI a second reader? Is it a triage? Is it autonomous? And so on. And let's think about what has changed with AI over the decades. What do we have now? We have faster computers, larger data sets, more advanced algorithms, especially those including deep learning. We've realized that there's a lot of other aspects in medical imaging interpretation that could use AI and incorporate it in clinical practice. That is, there's more clinical questions that AI might address. However, there's a lot that's the same over the past decades and that have been used. AI has been in medical imaging for at least 3 decades. They're the same clinical task of detection, diagnosis, response assessment. We have the same concern for garbage in, garbage out, and also the potential for misuse. The statistical evaluations that were there 3 decades ago hold now. And we also realize that a sufficient number of cases are needed, not just to increase the numbers, but to span the distribution of the disease and also the normal presentations. And as always, this is a multidisciplinary effort. So I'm going to go through 2 use cases that we've worked on in the lab and then talk about data repository effort. So a lot of our work is in AI and distinguishing between cancer and non-cancerous breast lesions, often on breast MRI. We do 2 ways. We look at it through the human engineered AI and also deep learning. For the human engineered, we have to worry about segmenting the tumor and extracting features. You can think of this as a virtual biopsy of the tumor. We segment the tumor. In this case, we're using fuzzy C-mains classifier to do that. We extract various features as shown here. Even though people might say I have 400 features, often you can reduce those to a very limited number of categories. We have hundreds of features we could pull off, but they relate to these 6 main categories. We pull them off, we can merge them by a classifier to yield signature indicating the likelihood of malignancy. This virtual biopsy, it's automatic, it's 3D, it's non-invasive. Roughly, it is a dynamic contrast enhanced MRI. It covers the complete tumor and you can repeat it, and I'll talk more about that later. We also look at deep learning. We are aware that you can do deep learning from scratch, though we use transfer learning. We do both feature extraction for the transfer learning as well as fine tuning. The example I'm showing here is using transfer learning for feature extraction. Advantage is we don't have to worry about lesion segmentation or extraction of features such as the size and shape. We just input the pixel level data to our deep learning approach. And we've gone into these details. There's a lot of papers. I don't have time to go into it here since I only have 20 minutes. But what I want to get across to you are the different ways when you're using multiple techniques. And I'm a big believer that we need to continue to look at the human engineered approach as well as deep learning approaches. So here I have a database of, I think, around 700 breast cancers. Red is a cancer case, green is a non-cancer. This is the base. You can look at this and think of this as an estimate of the likelihood of malignancy. This axis shows it for output from the human engineered AI, and this axis looks at it from deep learning. In a perfect world, we would have all the green down here and all the red up here, but you can see they span. Sometimes the human engineered gets it right and the computer deep learning doesn't, and also the other way, and sometimes they both get it wrong. But you can see by using both, kind of put a dividing line here, you could probably do better. And that's what we found. We looked at digital mammography, ultrasound, and dynamic contrast MRI of the breast in this task of distinguishing between cancers and non-cancers. Here we have the human engineered performance, and here the deep transfer learning performance. This is the area under the ROC curve. 0.5 is guessing, 1 is perfect. They're basically very similar, not statistically different, but when we just simply merge them, fusion of them, we get a statistically significant improvement in the performance. So we've continued down this path to continue to look at both methods. Another thing I want to point out is the garbage in, garbage out. Think about what you're inputting. What is the task? And in my lab, I like to say, think like a human. So here are 3 ways to present a breast MRI to a radiologist: the second post subtraction image, and the MIP. Radiologists often look at the MIP image. So we looked at these and input them into our deep learning method. We also focused, because this is a diagnostic test, we focused on the area around the tumor and not the entire breast. As with humans, the computer also preferred the MIP, the maximum intensity projection image. We've also looked at how do radiologists merge data when they have multi-parametric MRI, DCE, T2W, DWI. And we've looked at this using a radiomics approach, where we've looked at, do we fuse the features or do we fuse the output from the classifier? And both methods do give an improvement. But we can also extend that to deep learning, where here we have a DCE and a T2W. We can now merge using a classifier fusion, feature, merging the features from the DeepNet, or using the input to fuse the image data prior to going through it. By merging them, as expected, they all do better. And this is only done on about 800 cases, 1,000 maybe. But you see, it's still important to continue to look at the other input methods. DCE on MRI does do the best as alone, but do we get an advantage? Just like radiologists look at multi-parametric MRI, so can the computer. So we continue to do that. Now I'd like to mention to you that think about what happens when a woman comes in for screening. She may have a diagnostic imaging exam done if something suspicious is found. It's at that point there may be a biopsy done that yields molecular and genetic data. You can think at this point that you can do discovery. You can relate the quantitative analysis, your radiomics or deep learning of your imaging, and map it to the molecular information, genetic ones. And we've done that with the TCGA data. And if you find a signature here, you might then be able to move it to screening or risk of recurrence, do predictive modeling to get these virtual digital biopsies. But I like to point out that that signature that you might then pick off on a screening or risk of recurrence imaging, this virtual biopsy, is, we want to use virtual biopsy when an actual biopsy is not practical. You're not going to do actual biopsies during screening. We've extended our work to look at response to therapy and risk of recurrence. Here's an example. This is interesting because we looked at early on images, that is before treatment and after just the first treatment round. And our most enhancing volume predicts the recurrence-free survival. What's interesting, this is a single radiomic feature that has held and done better than merge in a lot of the other features. So don't always get too complicated. Look at individual features, human engineered and deep learning transfer ones. Working with the Cancer Genome Atlas and the Cancer Imaging Archive, we related our radiomics features to various cancer pathways, and we found that the transcriptional activities of various genetic pathways were positively associated with tumor size, blurred tumor margin, and irregular tumor shape, and that microRNA expressions were associated with the tumor radiomic phenotypes of size and enhancement texture, suggesting that microRNAs may mediate the growth of tumor and the heterogeneity of angiogenesis in the tumor. So now let's flip to COVID. I have, I think, 7 more minutes. The focus here is again on clinically and routine obtained images. And the COVID-19 pandemic presents a pressing important public health needs. We need to look at methods for surveillance and early detection, for triaging and differential diagnosis, as well as prognosis and prediction and monitoring of response to therapy. And it's still unknown what is the best route. And many are looking at chest radiography and CT to understand their role in the patient management of COVID-19 patients. A lot of research is done. As of October 23rd, there were over 6,000 papers on AI and imaging of COVID-19. Roughly half were peer reviewed. And we've also been doing that work. Here, Jordan Furman, a graduate student in my lab, has successfully developed a method using cascaded deep transfer learning on thoracic in order to distinguish which patients would be recommended for steroid treatments and which ones would not be. And Isabel Hu, also in my lab, a graduate student, is looking at chest radiographs in both predicting COVID-19 as well as in predicting the need for whether or not the patient should be put into intensive care or not. And here she's showing it with some heat maps from the grad camp. And this leads me to MIDRIC that many of you may be interested in. This is our website. This is a new NIBIB contract. We've been up for about just 2 months now. I'm the PI in it, but it's really led by 3 organizations, the APM, the RSNA, and the ACR. It's hosted at the University of Chicago and funded, and you can go to our website here to see it. It's a multi-group 2-year funded project. We're developing an imaging and data commons to have an intake portal through RSNA and ACR with various repositories and registries, and a public access portal on the UChicago Gen 3 data ecosystem. This is being done to address COVID-19 and for rapid, flexible collection, AI research, and dissemination of imaging and associated data. It also includes funded research projects to expedite the translation of AI from scientific findings to public dissemination and clinical benefit. So the 2 major components is to create this open discovery data repository, which will be of high image quality and quality assurance, and also to fund 12 machine intelligence projects to yield clinically relevant algorithms and software tools. Currently, we have 23 organizations involved, spanning the imaging expertise and well as the country. We start here. Data will be coming in through ACR RSNA, will be going under various stages of anonymization, QC. And so we have these two input portals, as I mentioned, ACR and RSNA. And we're also taking in the data currently be collected by the TCIA that many of you may be familiar with. And here is the RSNA import portal called Record. And I know you have access to these slides later. Here's the website and also the ACR research registry for COVID. Once they're in, we have methods being developed for assessing the data quality as well as the image quality. And we also have these various projects funded to look at, for example, how do we do task-based data distribution? What is the virtual clinical trials efforts? How can we use this for data science challenges? And last, we have a distribution portal. This is a public one, the public front door. It's hosted on the U Chicago Gen 3 data ecosystem, and we should have cases up very shortly. Gen 3 is the data commons, and they have hosted other ones. They host the NHLBI Biodata Catalyst, as well as the NCI Cancer Research Data Commons. So we're hoping by being there also we'll be able to crosstalk with some of the other registries. In fact, we will. It's not a matter of hope. I want to point out that MIDRC is being developed by the different organization taking different leads to create the discovery platform, the implementation platform, to conduct quality assurance and evaluation procedures, to distribute it on a world-facing data commons portal, and to link it to other clinical and research data registries. Also, through these three organizations, we've created 12 research projects that were immediately funded to expedite the AI research for rapid translation. So I'm going to end here. I believe my time is just up at 20 minutes. I want to remind you, whether it's breast cancer or COVID, there's many different clinical questions that need to be answered. One needs to make sure that one understands what the AI algorithm is, but also very important in the context of how it's going to be used. I like to end by thanking my lab. I've had many graduate students. I have 4 currently now. Everyone in the lab is brilliant. Research folks, as well as various collaborators. Here we are pre-COVID, and here we are now. Thank you.
Thank you so much. Fantastic talk. Yeah, if there's any questions, please enter them into the chat, and I guess we'll ask you, Mary Ellen, to answer them at the chat if possible. Suzanne, I guess it's break time right now, is that right?
Yeah, I think we're actually running over, so we'll go ahead and just invite folks to, certainly excuse yourself if you need to take a break, but I think we'll just go ahead and move on. We've got Dr. Bao, who is on the call and ready to do a talk as well. So we can go ahead and move forward if that's okay with everybody. So Dr. Bao, I'll go ahead and invite you to unmute yourself and share your screen. And Dr. Bao is from UTMB in Galveston. He's going to talk to us today about clinical applications of convolutional neural networks for COVID-19 chest X-ray screening. And go ahead and unmute yourself, Daniel.
Yeah, I'm a medical student, so I don't think I qualify. But I guess after 2 more years, I would be worthy of that term. Thank you for that introduction. So yeah, I am a second-year medical student. I'm talking about chest X-rays and the use of chest X-rays that are COVID positive for potential screening purposes. And I did this work with Dr. Peter McCaffrey, who's also at UTMB. He's both a pathologist and in the Department of Radiology. And so to begin, I want to discuss some of the clinical workflow aspects of COVID. And so as you probably are familiar with, you want to get a swab. That's a gold standard that looks for the RNA of the virus. And if you're symptomatic and you show up to the emergency room of urgent care, they're going to get you a blood saturation oxygen. So they're going to see if you're hypoxic. And then they're also going to take a chest x-ray most likely. And that's going to try to see if you have signs of pneumonia, because COVID, you can have asymptomatic COVID, you can have mild COVID infection that might not show up on chest x-ray. And so, and it's one of the cheapest ways to get a pretty good outlook on how severe your clinical features might be. There's some other things that you might know, if you get an antibody test, but those are not really used for screening. They're mostly used to see if you can be infected again. And so that's something that we want to keep in mind. This is the background that we're working with for COVID chest x-rays. And with the pandemic going on, there are tons of COVID positive chest x-rays, and they're from all around the world, different kinds of hospitals, rural or in a big metropolitan area. And a lot of these x-rays are not available for our public use, unfortunately, because they do have private health information. They require a lot of hoops to jump through if you want to get through them, but there are enough places out there and there are enough cases out there that they have some databases of COVID positive x-rays. And just for us data scientists out there, they're on GitHub, so you don't have to do some fancy password protection, or you don't have to go through other hoops. And they have some nice metadata, so they have their admission date, they have the radiologist note, they have the patient demographics, and those are all important when we start to use these x-rays, because those are things that we want to take into consideration when we're picking which ones to do our training on. And when we consider x-rays, and especially when we're considering them as disease-based x-rays, not normal-based x-rays, we want to understand that these features are transferable, because if you have COVID on one side of the world and you have COVID down the street here in Houston or Galveston, they should show up similarly. And that's something that we want to tackle with a transfer learning problem. And this image right here is actually normal. A lot of people have a lot of radiologists, and even as a medical student, I'm not that good at interpreting these images, but it's nice to have some labels associated it. And this one is actually normal. This one be counted as COVID positive, but ideally in a transfer learning situation, doesn't matter if this came from down the street where it's a normal x-ray, or if it's from across the world. And so these public databases can kind of be a way to test our transfer learning if we train on these public databases, and then see what happens when we feed in our own institutional images that no one's really seen before. But I mean, of course, if you show a radiologist, their transfer learning is probably good enough such that they can identify COVID positive chest x-rays regardless of which hospital they're at. And so we have a standard kind of pipeline here to do a convolutional neural network. We use a UTMB data set as a validation set, and we have the public image data set for just COVID positive as our COVID image source. Now we also have normal lung x-rays, and those are from the Kaggle. There's a competition. They also have public data available. And then for the pneumonia, for bacterial pneumonia, there are some slight differences between viral or COVID pneumonia versus bacterial pneumonia. Those are more low-bar consolidation, as you might hear the term. And so those are from the NIH. They have a big data set that's openly available. And we're kind of, it's almost like a Frankenstein little mishmash of data, but since they're all very good sources, they kind of provide the backbone of what we're trying to do in our normal public. If it's a true public data set, everything has to be on the open web. And you can see that there's a big data imbalance. So we're doing some undersampling in order to correct for this data imbalance. And we're just giving a one-hot label of if it's a normal or a COVID positive or a pneumonia chest x-ray. And the results of this are not so good. We're just testing on COVID-19 positive patients from UTMB. There wasn't a good way to request x-rays of normal patients, unfortunately. And we did some normalization because they're all from raw DICOM PACS images. And a key feature that I want to highlight here is that we only took one X-ray per patient, because when you're hospitalized, you get an X-ray almost every day. And that's something that can skew your data, and that can provide either duplicate data if you have similar X-rays day to day, or can provide incorrect data if your first day and the first day of your admission, you got an X-ray and they don't really find anything, but you have a positive COVID test. So that's why they put you under clinical observation. And you know that X-ray doesn't have that many features that might be present for pneumonia. And so that's not a good way to test on that. So we don't want to miss these patients that are actually normal. That's probably not good for their health, and it's not good for the healthcare system overall. And it might actually be okay if we misclassify them as pneumonia positive, because sometimes these things start to get pretty similar. If you have complete opacities throughout your whole lung, they appear similar to what COVID pneumonia looks like. And in order to address this low accuracy, we kind of just added all the data together. And that's what you would do normally in a pipeline if you didn't want to have this specific transfer learning problem. And so we added all the images together, and it's a lot more balanced, and it's still the same network architecture. It's still the same, one-hot labels. And we just did an 80-20 split, and the results are much better. And so this kind of looks at what you usually do for these kind of machine learning models. As I say, the data you train on needs to be very generalizable. It needs to be very, I guess, heterogeneous. Otherwise, if you show something that it's probably never seen before, it probably doesn't do too well. And on the UTMB data sets, even though they are still legitimate chest X-rays, they don't seem to recognize it as much. Maybe there's some machine differences in there, but there might also be some other factors, because we just have a better balanced data set. We have around 500, 500, 500. So that's one of the big things that you want to keep in check when you're training these models. And the biggest thing I take away from this result is that even with a lot of little tweaks and modifications and optimizations, I still can't reach a lot of the stuff that's currently out there on the literature. People have preprints about this. People have conference papers about this. They report over 90% accuracy on just COVID sets. So this is like 88% accuracy. This is all right. I'd say radiologists in real life probably have more than 90%, and you'd want a model like this to have more than 90% accuracy. And this is the best I could get. And unfortunately, it wasn't able to replicate some of the other ways from just the public data. And so this is using combined data, but just the public data, it's not, it's pretty hard to reach those numbers. And I kind of mentioned this before, but this kind of shows that the transfer learning is not feasible in the sense with the current amount of images that we do have in the public for COVID chest X-rays. And so this is one of the first works that has looked at this. Most of the other works that do have COVID images, they kind of end up doing the same thing. They just add all the data together. They use public data together, and they get okay results. But trying to make sure that your own institutional images are good. It's kind of like whenever you have laboratory samples, each hospital has their own standards and reference ranges for different laboratory samples, because they understand that there are slight differences in how they run these tests. And ideally for X-rays, you shouldn't have these differences because the patients are getting the same disease. And I think a lot of the other ways that other people are getting better results are potentially because they had multiple chest X-rays for each patient. So if you have 10 X-rays for a 10-day stay, and you're trying to add that model, you're trying to use those images to train a model, and you have an 80-20 split, 2 of those X-rays are going to be in your testing data, and you've already seen them in training 8 times. And so it's not very good for a good model training. And it kind of really shows that not many people are using this as a true screening tool, in the sense that you could give everyone a chest X-ray. Unlike for other diseases, for tuberculosis, you could do this for screening, and the numbers aren't good enough. 88%, you're missing quite a few people if you want this to be a very good screening procedure. However, it might be better as a severity measure, because As we do note, if you're throughout the hospital stay, you do find radiologic changes, and that's something that can be noted. And that's something that I want to bring in to future works, which is implementing more data from the electronic health record. And that would help with not just having a positive test result as the main label, and having more things that we could use to expand the data and possibly move on to segmentation for more relevant areas that are closer to COVID. So this was not a true model where you would replace a radiologist. It could aid the radiologist in identifying areas that they need to look at again. And with a lot more images, you might want to consider some other methods like semi-supervised or self-supervised learning. And I think one of the other ways to increase your data robustness is to generate artificial COVID-19 positive images. And that's kind of something that you would do to supplement limited data. And I think that's all I really have. If you guys have any questions, I'll open it up. And these are some of the works that I looked at before I started on this project. And if you aren't able to type out the question, you can contact me down below.
Thank you, Daniel. We'll invite you to jump over to the chat. If anybody wants to enter questions, they can enter them for you there.
So hello, everyone. This is Jiang Guo, a graduate student from the Department of Bioengineering, Rice University. Today, I'm glad to talk about my project using deep learning to quantify bacterial self-organization patterns. So thank Chris for his help on preliminary model building. Thank Jessica and Roy for their work in wet lab. We have collected a lot of data from there. And thank Ankit and Oleg for their mentorship on deep learning and system biology. If you're interested in my research project, you can visit these websites. To begin with, let me show you our research target, Myxococcus densis. The myxo is a kind of bacteria fed on other bacteria, like E. coli. On the left, we can see how they prey on an E. coli colony. Under nutritional stress, the Myxococcus densis initiates a developmental program to self-organize into fruiting bodies, large mounds in which cells differentiate into metabolically inert spores. You can see these round spores forming from left to right on this plane. These spores are resistant structures used for survival under unfavorable conditions. It is interesting to see that single cell creatures can form such complex structure cooperatively. In order to better study this phenomenon, we have to quantitatively analyze the features of the self-organized procedure. There are two key features. One is the aggregation, including the cell density, the size, and the shape of the aggregates. The other is the streams connecting different aggregates. The cells move in and out of the aggregates in the streams. They show how aggregates are formed and collapsed. We have two different kinds of microscopic views. One is the conventional phase contrast image. The other is a TD tomato fluorescent image. The phase contrast images are easy to capture, but as you can see, there are a lot of artifacts, such as the reflection ring, than the bottom right. It is hard to use the phase contrast image to do analysis, so we use the TD tomato fluorescent images. If we normalize the TD tomato image with different methods and zoom in, we can get different levels of information. On the top right, you can see the TD tomato image with interpretable aggregation information. You can tell the aggregation size, shape, and the cell density. On the bottom right, you can see the streams connecting different aggregates. The stream information is much clearer than the top image. Therefore, we know that the fluorescent image is useful for us to study the aggregation. However, it is hard to produce. We have to use the transgene technology to introduce a new fluorescent protein. We need much labor work. and the fluorescent protein may be toxic to the cell and affect the cell behavior. The phase contrast images are easier to produce, but are hard to use. So is there any way to transform between fluorescent and conventional microscopy to better quantity aggregation? The models built for image cell transfer problem may answer our question. Before we train our model, we have to normalize the data and make the images compatible with our model. First, we use Clay-He and Naxos methods to make the contrast consistent. Then we crop, flip, and rotate the images to get smaller 256 times 256 images. These images are big enough to contain at least one aggregate and small enough to train our model. If we use the whole image in a deep neural network, it may exceed our memory limit. Then we replace the Clay-He with He to normalize the image and get cell stream information. Now we have one input and two ground truths. Clay-He image for aggregation information and He image for stream information. Next, we build a pix-to-pix HD model. This model has two levels. First, we downscale the input image and train the small convolutional residual network to produce a downscaled fluorescent image. We then train the large model with the information already learned in the small model by adding the second last feature layer to the middle. The original single output model has one output. We use a multi-layer discriminator and feature matching loss to train our model. Here, if we train the model on this single image, where the stream signals are weak, the model cannot learn the stream information very well. Here is the result. After training the model for 3,000 epochs, the single output model can recover the aggregation information, such as the cell density, aggregate shape, and size pretty well. However, when we apply He on these images and try to recover the stream information, we find that some areas are blurry. It is possible an overfitting of the weak signal. Is there any way to improve our model? Then we design this two output architecture. So it's almost the same, but has two outputs. Most of the hidden layers are shared so that the model is learning both aggregates information as well as the stream information during training. The details are learned through this pathway, but then be passed to the other output so that this output may contain both aggregate information and detailed stream information. As a result, we can still recover the aggregate information very well. What's changed is that the stream information is well recovered. There is almost no blurry area in our generation, which means that with the output model, the aggregation information and detailed stream information are well recovered. So this is qualitatively evaluated by human eyes, how to evaluate the results quantitatively. So this is the log mean square error used for evaluation. We first subtract the generation from the real image and calculate the absolute value. So this image shows the difference between the real image and generation. Next, we calculate the square values on each element. Calculate the average and logarithmic. We get the value minus 3.6. This value represents the difference between real image and generation. The smaller, the better. Then we put the log mean square error into the time scale. The time scale represents images taken in a time series. For example, the point 100 minus 3 means that if we compare two images of the same microscope view, but are taken 100 minutes apart, the log mean square error is minus 3. So since the cells are moving, the larger the time difference, the higher the mean square error. The log mean square error between our generation and ground truth is shown as the dotted lines on these figures. On the left, we evaluate the aggregation recovery. The generated image is more similar then the images taken about 40 minutes apart. On the right, we evaluate the stream information recovery. The generated image is more similar than the images taken 30 minutes apart. So the two output model outperform the one output model on both aggregation recovery and detailed stream recovery. Finally, we want to see how our model performs on the movie transformation. We can track the movement of some single cells in our generated movie, while the stream is almost unable to capture in the original phase contrast image. However, our model has its weakness. The generated movie is not as continuous as a real movie. Perhaps the nearby frame assisted model can help with the movie generation.
2 minutes.
Then we come to the conclusion. We have developed a two output model that can help us transform phase contrast images to fluorescent images with both aggregate information and detailed stream information. So the next step, we're going to do the reverse transformation to transform the fluorescent image back to phase contrast image so that we can generate phase contrast images from the computer synthesized fluorescent images. That is all of my presentation. Thank you for listening. Any questions?
Thank you very much. Great talk. So we invite our audience to enter any questions you may have in the Umbrella chat, and we'll invite you to jump over there and answer those directly there. I see there was some interaction with the previous speaker, so thank you so much. Great talk.
Thank you.
So for this next session, what we're going to do, this is our rapid fire talks, which will take us into our networking session that we'll do afterwards. So what we've done is we've invited the rapid fire speakers to submit 3 slides. They're going to do short 3-minute presentations. Dawn, if you want to go ahead and pull up those slides to share. Perfect. All right, so again, you'll have 3 minutes, and we'll go ahead and start with Ketan. You can unmute your microphone and turn your video on. Ketan, can you hear us?
Sorry about that.
There we go. Perfect.
All right. Good afternoon, everyone. My name is Ketan Gagda. I'm a faculty member in the Department of Radiology at Textuals and Baylor. My research interests are in the development of novel imaging agents and imaging methodologies for early detection and monitoring disease progression, as well as prediction and monitoring of treatment outcomes. We are applying these novel methods as well as imaging agents in a variety of diseases, including Alzheimer's disease, cancer, cardiovascular, and placental imaging, with the ultimate goal of translating these technologies to address unmet clinical needs. Here are examples of images from 2 projects. One involves the development of an MR molecular imaging agent for the detection of amyloid plaques in Alzheimer's disease. The agent is expected to enter phase 1 clinical trials next year. And below is a 3D segmentation of a rat's enteroplacental vasculature using a nanoparticle contrast agent that we developed to enable accurate detection of placental pathologies. In the next slide, I would like to briefly share a relatively new area of research where we are using nanoradiomics, a term we coined to describe high throughput extraction of quantitative features from 3D in vivo images of intratumoral nanoparticle distribution maps in order to probe the tumor microenvironment. In a recent study, we showed that nanoradiomics detected treatment response to tumor microenvironment directed cellular immunotherapy that was otherwise not feasible using conventional imaging-derived tumor metrics. We are also beginning to apply machine learning methods to further augment the nanoradiomics platform for tumor profiling. Hopefully we'll have some more interesting data to share in the symposium next year. That's all I had to say for today, so thank you for your attention.
Thank you very much. We'll move on to Rob.
Oh, I thought I was third, but I'm here. Okay. So, yeah, I'm Rob Raphael, Associate Professor of Bioengineering at Rice. Our imaging is, this conference is a little bit new for me. I normally am doing membrane biophysics, transport, and the auditory vestibular system. About a year ago, we got into optical coherence tomography, and so this is a little bit of a different technique than you've heard today. OCT lies in this sweet spot where you can get imaging depth of up to almost a centimeter and keep your resolution of about 1 to 10 microns. So we do both OCT and computational modeling in the inner ear. We're kind of seeking collaborators who might have expertise in imaging analysis, segmentation, and machine learning to kind of these techniques have not been applied to OCT images so much to my knowledge. So let's go to the next slide. Yeah. Okay, good. So one of the challenges of the inner ear is that it's encased in the temporal bone, which is the densest bone of the human body. So you need to also study the tissue in situ, because if you break in the bone, you are going to break up the ionic gradients, which are responsible for keeping this tissue functioning. So that's why OCT is our method of choice. Most of my career, I've studied the cochlea. More recently, we're looking at the vestibular system, which is responsible for our sense of hearing and balance. And it basically, there's a lot of diseases, for example, Meniere's disease, which causes attacks of vertigo, for which there are no effective clinical treatments. And we believe that that's the case because there's just little understood about some of the molecular mechanisms that may cause some fluid disturbances in the inner ear. So the real barrier is that we can't non-invasively image this at the resolution we need to kind of understand both the normal and pathological function of the tissue. So next slide, yeah. And I'll just show you this little picture of our OCT machine. We listed the advantages here of OCT. 1 minute, yeah. So I'll just show you these two images. One in which is in case in which we've dissected a mouse and exposed one of the semicircular canals on the bottom left there, and then you can see through the bone to the intramembranous compartments, which are two fluid-filled compartments known as endolymph and perilymph. And then on the right, what we did is we isolated the whole temporal bone out of the animal. And here we're using three-dimensional imaging to kind of see what the structure of the semicircular canal is. And on the right there, we labeled two compartments. It's not high enough for you guys to, I can't see it on my computer, so you guys probably can't see it either. But we identified the two kind of end organs known as the utricle and the saccule. And these are what basically where the sensory hair cells are that sense the fluid motion. The other thing OCT can do is Doppler interferometry. So it can also enable us to measure vibrations in these compartments. So I'm done. Just on time, Susan. Good.
Okay, thanks, Rob. Now we'll move on to Elijah Rockers from Methodist Hospital Research Institute.
Hi there. Can you hear me okay?
Yes, we can.
Wonderful. I'm sorry, I don't have a webcam actually in my office, unfortunately, so you'll just have to make do with my picture up there. But as Suzanne said, I'm Elijah Rockers. I'm a analyst over here at Houston Methodist at the Translational Alzheimer's Center. Basically, we are a clinic, so we see patients daily. And my PI, who is the director of the lab here, also participates in a lot of research studies. So we do clinical trials with pharmaceutical companies, and we also, through philanthropy, are able to do a lot of studies of our own. And so basically, my role is to analyze and process the MRI and PET images. And PET is one of the strengths of our lab here. We have a cyclotron here in the building, and so we're able to produce compounds which have a much shorter half-life than fluorine-18, such as carbon-11. So we're able to do, some tracers that tend to have a little bit more specificity as far as the targeting goes, and in particular, we look at Alzheimer's and other types of dementia, as you can see on the screen here. And so basically, we do a lot of multimodal sort of studies where we're looking at the deposition of amyloid on the brain and also the tau with F18, AV1451. And we're looking at metabolism there with FDG, and then we use MRI to look at cortical thickness. We do a lot of resting state MRI and diffusion tractography. And basically what we're trying to see is how the disease progresses among these different types of dementia. And so while we do look at a lot of Alzheimer's, we're also really focused on other types of dementia which are more focal, and we can sort of get a better handle on the progression, how these diseases might progress, whereas Alzheimer's is typically a more diffuse kind of disease that's a little bit harder to pin down like this. And so one other thing that we do very well here that not a lot of other sites do is arterial line sampling. So when we do PET, a lot of sites will have to use a reference region or something like that. So they're basically making assumptions about the amount of binding that happens in these sites. And unfortunately, a lot of these studies can be a little bit quantitatively off a little bit because they're not doing exact quantification, which is something that arterial line sampling addresses. Unfortunately, it can be kind of an awkward protocol for the patients to go through because this isn't just a simple venous thing. This is directly into the arteries. So it can be a little bit of an awkward thing, but luckily here at Methodist we have anesthesiologists and the whole medical team to deal with that. And so can you go on to the next slide, please? Okay, and so also, since this is an AI and machine learning conference, I just wanted to talk a little bit about more what I've been doing sort of on my own with the lab here. And that's looking at segmenting MRI, especially with structural T1 and T2 flare for white matter hyperintensities. I believe Dr. Gaber was one of the speakers earlier, was looking at multiple sclerosis. And so this is kind of a similar idea here. We have, extensive lesion damage, and we're basically comparing several different types of methods that you see here, such as the popular UNET, which we've heard a couple of times today. And so we're basically segmenting these two types of images and comparing, how they compare across these different modalities and what that might mean physiologically, as well as just the performance of these methods. And then final slide. And so basically, I was just trying to introduce myself, let you guys know who I am. And so some other things that I'm interested in here. As you can see, I already mentioned resting state, and I think Dr. Kaleholtz was one of the early speakers today who was talking about dynamic functional connectivity, and that's something I'm really interested in, especially when it comes to Alzheimer's and cognition. And so with that, I think I'll yield my time. Thank you very much.
Thank you, Elijah. We appreciate it. Okay, we'll move on to Kalina Slavkova from UT Austin.
Hello. Hi, everybody. My name is Kalina Slavkova, and I'm a PhD candidate in the physics department at UT Austin. I work with Thomas Yankulov in the Center for Computational Oncology, for which you can find more details at the website here, cco.odin.utexas.edu. My research interests are in developing acquisition and analysis methods for dynamic breast MRI data for quantitative imaging in the abbreviated setting. We have a high temporal resolution breast DCE MRI dataset here at UT, with which we've been testing our deep learning reconstruction scheme and various perfusion model analyses as well. We are interested in raw case-space data from high spatial resolution DCE MRI datasets to better measure the ability of our deep learning scheme to capture high frequency information. This top panel here is an example slice from our UT DC MRI dataset with a 7.27 second temporal resolution. This bottom panel here is a parameter map of that corresponding slice from our perfusion model analysis. My PhD work is split up into 3 aims, 2 of which I'll mention here. In my first aim, we investigate the possibility of quantitative analysis of abbreviated breast DCE MRI. We start with something called an FTC, the full time course, and then we truncate it into a series of abbreviated time courses, which contain the first n number of time points from the FTC, where we specify a set of n's. Then we fit the full-time course to the Ketty-Tofts perfusion model, and then the abbreviated time courses to the same perfusion model. From this analysis, we get model fits and parameters distributions over the tumor regions. Lastly, we compare the FTC and ATC parameter estimates on a voxel-wise basis by computing the percent error and the concordance correlation coefficient. We've seen some promising results, and we're currently writing up this work. My aim 2 investigates whether deep learning can help us acquire high spatial and high temporal resolution DCE MRI for our quantitative analyses. We start with a set of Fourier data, coil sensitivities, and we have our ground truth images. We have an MRI specific forward operator, A here. Oh, sorry, I wasn't saying next slide. I'm on my 3rd slide now. Yes, yeah, oops. Yeah, sorry about that. So now I'm on my aim 2 slide. As I was saying, we have an MRI specific forward operator here, which includes a sampling mask, the Fourier transform, and our coil sensitivities from our 16-channel coil arrays. The specific architecture we're working with is the conv decoder by Heckel et al. In this 2020 publication. We then perform this minimization over the weights of the neural network in the supervised and the self-supervised setting. Lastly, we have the final output here in the bottom 2 panels in the bottom right corner of the screen, which we compare to our ground truth image. And after performing perfusion model analyses, we compare that to the ground truth results as well. So thank you all for your time. Apologies about the slide transition.
No worries. Thank you, Kalina. Impressive talk. So we've finished up a little bit early because we had a couple of our rapid fire speakers have to back out at the last minute. But I do want to just thank all of you for attending today. In a second, I'll give you some instructions for the next part of our meeting, which is an invitation to utilize the networking functions of the Brella platform. Again, I just want to say thank you to all of our speakers, all of the organizers, and of course to all of you. I do want to mention, I mentioned it at the beginning of the day, that the Translational Imaging Cluster hosts a monthly seminar series on the 3rd Wednesday of the month called Hot Topics in Imaging. This particular month, we're going to hear from Dr. Irina Larina from Baylor College of Medicine. The title of her talk is Shining Light on Early Development. And that's going to be November 18th between 1 and 2 p.m. If you'd like more information on that, you can contact me at the GCC. And then you're also going to be receiving a survey. We want to hear how well you were able to network using the Brella platform, what you thought of the talks, and how we can improve moving forward with our conferences. Just a little bit about this next session of networking. If on the left side of your screen, where you saw the stream tab, there is a people tab, and you can scroll through the people that are in attendance at today's conference and click on their name, and there is a function there where you can request a meeting. Once they accept that meeting, you can either enter into a room to start that meeting and chat with them. In the upper right-hand corner, there's also a camera icon that you can click on. And that will enable you to be able to have kind of a face-to-face, at least in this virtual world, video meeting with them. Again, we thank you so much. We hope you'll take a few minutes to take our survey. You'll receive that tomorrow. And we invite you now to just reach out to somebody that you heard from here today that you might be interested in collaborating with in the future. Thanks again.